Hey there, Night Wanderer. Welcome back. Before you settle in tonight, maybe adjust your pillow, pull that blanket a little closer. Good. Now, let me ask you something. Have you ever felt like something else was making decisions for you? Like some invisible force was guiding your thoughts, your cravings, your impulses, that strange pull toward the kitchen at midnight, that sudden urge to change your entire routine? Well, tonight we're going to explore something that might make you reconsider what control really means. Because out there, in the natural world, there are creatures so small you'd never see them coming, yet so powerful they can rewrite the very thoughts of their hosts. Parasites, mind controllers, the puppet masters of the biological world. And here's where it gets interesting. Right now, as you're lying there listening to my voice, there's a decent chance you're carrying one yourself. Don't worry. Take a breath. Most are completely harmless, but some can change behavior in ways that seem almost supernatural. Before we dive deeper, if you're enjoying this journey into the hidden world of biology, go ahead and like this video. Subscribe if you'd like more of these late-night explorations into science and nature. And drop a comment below telling me where you're tuning in from and what time it is. Maybe share what usually helps you sleep. I always find it fascinating to see who's joining us from around the world, and honestly, it helps me know what to explore next. Over the next few hours, we're going to journey together through the fascinating, sometimes unsettling, always remarkable world of parasitic biology. We'll meet fungi that turn insects into zombies, worms that make their hosts seek water with desperate determination, microscopic organisms that might be influencing human personality right now in ways we're only beginning to understand. Now, I know what you might be thinking. Parasites? For sleep? But here's the thing. There's something deeply calming about understanding the hidden mechanisms of nature, about seeing how life, in all its strange and wonderful forms, has found ways to survive. These stories aren't meant to disturb you. They're meant to remind you that the world is far more intricate, far more interconnected than we usually stop to consider. And sometimes that perspective is exactly what we need to quiet our racing minds. Think about it this way. Every single day, your body coordinates trillions of cells, each one following chemical instructions so precise they make the most advanced computer look clumsy. Your immune system fights off invaders you'll never know about. Your brain processes thousands of signals every second, filtering out the noise so you can focus on what matters. You are, right now, a walking ecosystem, a universe of biological cooperation. And parasites? They've simply learned to navigate that universe in ways that benefit them. They're not evil. They're not malicious. They're just extraordinarily good at survival. Some of them have been perfecting their craft for hundreds of millions of years, long before humans ever walked upright, long before we built cities or wrote languages or wondered about our place in the cosmos. So go ahead and close your eyes if you'd like. Let your breathing slow. Feel the weight of your body sinking into whatever surface is supporting you. Notice how your shoulders might be holding tension. Let that go. Tonight, you don't need to be in control of anything. You can just listen. Just drift. Just learn. Because we're going to meet nature's most sophisticated hackers. The organisms that discovered how to reprogram life itself millions of years before humans ever dreamed of computers. We'll explore the elegant chemistry of mind control, the evolutionary arms races that have shaped entire ecosystems, and the surprising ways that parasites might actually be essential to the balance of life on Earth. The lights are dim, the world is quiet, and the invisible kingdom is waiting to reveal its secrets. Let's begin. So let's start with a simple question. What does it mean to be in control? Right now, you're choosing to listen to this. You chose to lie down, to close your eyes, to let your mind wander into the world of biology and sleep. Or did you? What if I told you that some of the choices you make, some of the preferences you have, might not be entirely yours? What if something microscopic, something you've never seen, has been quietly influencing your decisions for years? Welcome to the world of behavioral parasitism. This is where biology gets strange, where the line between self and other becomes blurry, where tiny organisms have learned to reach into the minds of their hosts and pull invisible strings. Now, when most people think of parasites, they imagine something unpleasant. Tapeworms coiled in intestines, ticks buried in skin, lice crawling through hair, and yes, those exist, but those are the amateurs. The parasites we're talking about tonight are far more sophisticated. They don't just steal nutrients. They don't just live inside a host and cause discomfort. They reprogram behavior. They change personalities. They make their hosts do things that serve the parasite's needs, often at great cost to the host itself. Think about that for a moment. An organism so small you could fit thousands of them on the head of a pin, reaching into a brain millions of times larger than itself, and changing how that brain makes decisions. It sounds like science fiction, but it's happening right now, in forests and oceans and cities all around the world. It's been happening for hundreds of millions of years. Let's start with one of the most famous examples. There's a microscopic organism called Toxoplasma gondii. It's a single-celled parasite, and it has a very specific goal. It needs to get inside a cat. Not just any cat, but the digestive system of a cat, because that's the only place it can sexually reproduce and complete its life cycle. But here's the problem. This parasite often finds itself inside a mouse or a rat, and rodents, understandably, are terrified of cats. They've evolved over millions of years to avoid anything that smells like a predator. It's hardwired into their brain. The scent of cat urine triggers an immediate panic response. Run, hide, survive. But Toxoplasma needs that mouse to get eaten, so it does something remarkable. It changes the mouse's brain. Specifically, it targets the amygdala, the part of the brain that processes fear and emotional responses. It alters neural pathways, it manipulates neurotransmitters, and slowly, over the course of weeks, that mouse loses its fear of cats. In fact, studies have shown that infected mice are actually attracted to the smell of cat urine. They seek it out. They linger in areas where cats have been. They essentially walk right into the jaws of their predator. The mouse isn't aware this is happening. It doesn't feel mind-controlled. It just feels like its preferences have changed. Maybe it's curious. Maybe that smell isn't so bad after all. Maybe it's worth investigating. And then, in a moment of fatal curiosity, the cat pounces. The mouse is consumed, and Toxoplasma completes its journey, finally arriving in the gut of its definitive host, where it can reproduce and send its offspring out into the world to begin the cycle again. From an evolutionary perspective, this is beautiful, elegant. The parasite doesn't need size or strength. It doesn't need claws or venom. It just needs to rewrite a few lines of code in its host's operating system, and it does this with extraordinary precision. Infected mice don't lose all fear. They're still afraid of other predators. They still avoid open spaces where hawks might spot them. The parasite only removes the fear of cats, because that's the only change that serves its purpose. Any broader alteration might make the mouse too reckless, too likely to die before a cat can eat it. So the manipulation is surgical, specific, almost respectful in its efficiency. Now, toxoplasma isn't alone. There are thousands of parasites that alter host behavior, each with their own methods, their own targets, their own evolutionary strategies. Some make their hosts more aggressive, others make them lethargic. Some drive hosts toward water, others make them climb to high places. Some even cause their hosts to sacrifice themselves in ways that seem almost intentional, almost purposeful. And here's what makes this so fascinating. These aren't random effects. These aren't side effects of infection, like a fever or inflammation. These are adaptations. Over millions of years, parasites have evolved the ability to manipulate behavior in ways that directly benefit their survival and reproduction. They've learned which neurons to target, which chemicals to release, which brain regions to colonize. They've become, in essence, neurological engineers. Let's think about what this means. Every behavior you see in nature, every action an animal takes, might not be entirely its own. That cricket jumping into a stream, that caterpillar climbing to the top of a tree, that bird sitting exposed on a branch, making itself an easy target. These could all be the work of parasites, invisible puppeteers guiding their hosts through a performance written millions of years ago. And the spectrum of control varies wildly. Some parasites cause subtle changes, a slight shift in preference, a minor alteration in routine. You might not even notice if you were watching the host, but other parasites take complete control. They override every instinct. They turn the host into something almost robotic, following a script that leads inevitably to death, but not before the parasite has achieved its goal. Scientists call this the manipulation hypothesis. The idea that these behavioral changes aren't accidents, but carefully evolved strategies. And the evidence keeps mounting. Researchers have found that parasites often produce molecules that mimic neurotransmitters: dopamine, serotonin, GABA, the same chemicals your brain uses to regulate mood, motivation, fear, and pleasure. The parasites have essentially learned to speak the language of the nervous system, and once you can speak that language, you can tell the brain what to do. Take a moment to consider the implications. Your brain, right now, is a symphony of chemical signals. Billions of neurons firing, communicating, coordinating. And all of it depends on neurotransmitters, these tiny molecular messengers that flow between cells. If something could alter those signals, even slightly, it could change how you feel, how you think, what you want. And parasites have been doing exactly that, perfecting their technique over evolutionary timescales we can barely comprehend. Now, before you start worrying, remember that most parasites are highly specialized. They've evolved to manipulate specific hosts in specific ways. A parasite that controls a mouse isn't going to have the same effect on a human. Our brains are different. Our chemistry is different. But the principle remains. The potential is there. And as we'll explore later, there are parasites that do infect humans, and some of them might be influencing us in ways we're only beginning to understand. But for now, just sit with this idea. Behavioral parasitism, the art of invisible control. It's not magic. It's not supernatural. It's just biology, pushed to its most intricate extreme. And it's happening everywhere, all the time, in ecosystems across the planet. a hidden layer of influence that most of us never think about, but that shapes the natural world in profound ways. So, the next time you see an animal doing something strange, something that doesn't quite make sense, something that seems almost self-destructive, ask yourself, is that really its choice, or is something else pulling the strings? So now you know that parasites can control behavior, but how? How does something so small reach into a brain and flip switches that evolution spent millions of years building? How does a microscopic organism override instincts so powerful they define what it means to be that animal? Let's pull back the curtain and look at the machinery. The beautiful, terrifying machinery of neurological hijacking. First, we need to understand what the parasite is up against. The brain, any brain, is arguably the most protected organ in the body. It's surrounded by bone, it's cushioned by fluid, and most importantly, it's guarded by something called the blood-brain barrier. Think of this barrier as a highly selective security checkpoint. It allows oxygen, glucose, and essential nutrients to pass through. But it blocks almost everything else. Toxins, pathogens, large molecules, they're all stopped at the gate. This barrier exists because the brain is delicate. One infection, one wrong chemical, and the entire system could collapse. So, for a parasite to control the brain, it first needs to get inside. And this is where things get ingenious. Some parasites don't even try to cross the barrier themselves. Instead, they send chemical messengers, molecular keys that can slip through the checkpoint and unlock doors on the other side. Others are more direct. They actually breach the barrier, using enzymes to temporarily weaken it, creating small gaps just large enough to squeeze through. It's biological lockpicking at its finest. Toxoplasma gondii, for example, forms what scientists call tissue cysts in the brain. These are small clusters of the parasite wrapped in a protective wall, hiding inside neurons. They can remain dormant there for years, even decades, quietly releasing proteins and other molecules that alter how those neurons function. The host's immune system knows they're there, but the cysts are remarkably good at evading detection. They've evolved to be invisible, or at least invisible enough. Once inside the brain, the real work begins. The parasite needs to change behavior, and behavior is controlled by neurons, those long, branching cells that carry electrical and chemical signals throughout the nervous system. But neurons don't work alone. They communicate using neurotransmitters, those molecular messengers we mentioned earlier. When one neuron wants to send a signal to another, it releases neurotransmitters into the tiny gap between them, called the synapse. These molecules drift across, bind to receptors on the next neuron, and trigger a response. Fire. Don't fire. Get excited. Calm down. The entire language of the brain is written in neurotransmitters. Now here's where parasites show their brilliance. They've learned to speak this language. Some parasites produce their own versions of neurotransmitters, flooding the system with signals that mimic natural brain chemistry. Others block receptors, preventing legitimate signals from getting through. And some do both, creating chaos in the neural networks that govern behavior. Let's talk about dopamine. This is one of the most important neurotransmitters in the brain. It's involved in motivation, reward, pleasure, and movement. When you accomplish something, when you eat something delicious, when you experience joy, dopamine is part of that response. It's what makes you want to do things, to seek out experiences, to move toward goals. Now, Toxoplasma gondii has been found to increase dopamine levels in the brains of infected rodents. How? It carries genes that code for an enzyme called tyrosine hydroxylase, which is a key component in dopamine production. The parasite essentially hijacks the brain's dopamine factory and cranks up production. Why would it do this? Because dopamine affects risk-taking behavior. Higher dopamine levels are associated with increased curiosity, reduced fear, and greater willingness to explore novel environments. For a mouse that needs to get eaten by a cat, this is perfect. The mouse becomes bolder, more adventurous, more likely to investigate that interesting cat smell instead of running away. The parasite doesn't need to understand neuroscience. It just needs to produce the right molecule, and evolution has spent millions of years refining which molecule works best. But dopamine is just one piece of the puzzle. There's also serotonin, which regulates mood, anxiety, and social behavior. There's GABA, which controls inhibition and calm. There's norepinephrine, which governs alertness and stress responses. Each of these neurotransmitters plays a role in shaping how an animal perceives the world and responds to it. And parasites have learned to manipulate many of them. Think about it like this. Imagine your brain is a sound mixing board, the kind you'd see in a recording studio. Each slider controls a different frequency, a different instrument, a different element of the final sound. Normally, your brain adjusts these sliders automatically, based on millions of years of evolutionary programming. Fear response? Turn up norepinephrine. Social bonding? Increase oxytocin. Relaxation? Boost serotonin and GABA. But now, imagine someone else gets access to that mixing board. They start moving sliders. They turn some up, some down, creating a completely different mix. The equipment is the same, the room is the same, but the output, the behavior, is entirely transformed. That's what parasites do. They don't need to rewire the brain from scratch. They just need to adjust the levels of chemicals that are already there. And because these chemicals are so powerful, even small changes can have profound effects. A 10 or 20% increase in dopamine might not sound like much, but in the brain, that's the difference between caution and recklessness, between fear and fascination. Now, different parasites target different parts of the brain depending on what behavior they need to change. The amygdala, which processes fear and emotion, is a common target. The hypothalamus, which controls basic drives like hunger and thirst, is another. Some parasites even affect the prefrontal cortex, the region involved in decision-making and impulse control. It's not random. It's strategic. Each parasite has evolved to manipulate the specific brain regions that control the behaviors it needs to alter. And here's something that might surprise you. This kind of manipulation isn't always harmful to the host, at least not immediately. Many infected animals can live relatively normal lives for months or years. They eat, they sleep, they reproduce. The only difference is that one specific behavior has changed. That cricket that used to avoid water now seeks it out. That ant that used to stay with its colony now climbs alone to the top of a grass blade. The rest of their life continues as usual, until, of course, the moment when that changed behavior leads to their death and the parasite's reproduction. There's a strange elegance to this. The parasite doesn't want to destroy its host prematurely. It needs the host alive and functional, right up until the precise moment when death serves the parasite's purpose. So, the manipulation is calibrated, measured, just enough to change the outcome, but not so much that the host collapses before completing its role in the parasite's life cycle. Scientists are still unraveling exactly how all of this works. New discoveries are made every year, new molecules are identified, new pathways are mapped. And with each discovery, the picture becomes more complex and more astonishing. These parasites aren't just dumping chemicals randomly into the brain and hoping for the best. They're conducting a precise chemical symphony, targeting specific neurons, specific receptors, specific moments in the host's life. And remember, this all evolved naturally. No intelligence designed it. No conscious mind planned it. It's just natural selection, rewarding parasites that happen to produce the right molecules at the right time in the right place. Over millions of generations, those small advantages accumulated, refined, perfected, until we're left with what we see today. Organisms that can reach into the most complex structure in biology, the brain, and rewrite its code. So when you think about consciousness, about free will, about the sense that you are the one making your decisions, remember this. Your brain is a chemical system, and chemical systems can be influenced. The boundary between self and other, between your thoughts and outside influence, might be thinner than you ever imagined. Next, we're going to meet the parasites that take this to the extreme. The ones that don't just nudge behavior, but completely override it, turning their hosts into something that looks almost like a puppet on strings. Deep in the rainforests of Thailand, Brazil, and tropical Africa, something is happening that looks less like nature and more like a horror film. But it's real, and it's been happening for at least 48 million years. We know this because scientists have found fossilized leaves with bite marks, death grips frozen in time, evidence of an ancient relationship between fungus and insect that has endured since before humans, before mammals dominated the earth, before the continents looked anything like they do today. This is the story of the zombie makers. Fungi that don't just infect insects, they possess them. And the most famous of these is a genus called Ophiocordyceps. The name alone sounds ominous, and what it does is even more remarkable than its name suggests. Let's start with a carpenter ant going about its day in the canopy of a tropical forest. It's foraging, communicating with its colony through chemical signals, contributing to the collective survival of thousands of its sisters. Everything is normal, until it walks across a leaf where fungal spores are waiting. These spores are so small, the ant doesn't notice them. They stick to its exoskeleton, then begin to germinate, drilling through the hard outer shell using a combination of mechanical pressure and enzyme secretion. Within hours, the fungus is inside. But here's what's strange. The fungus doesn't kill the ant immediately. Instead, it begins to grow, spreading through the ant's body, extending thread-like structures called hyphae through the muscles, the organs, the circulatory system, and as it grows, it starts to release compounds, chemicals that the ant's brain recognizes, chemicals that look and act like the ant's own neurotransmitters. The fungus is learning to speak ant. Over the next week or so, the ant begins to behave differently. It becomes disoriented. It wanders away from its colony, abandoning the cooperative behavior that defines ant society. It leaves the canopy, climbing down to the forest understory to a very specific altitude. Scientists have measured this. Infected ants consistently descend to about 25 centimeters above the forest floor. Not higher, not lower, 25 centimeters. Why? Because at that height, the temperature and humidity are perfect for fungal growth. Too high, and it's too dry. Too low, and it's too variable. The fungus needs precision, and somehow it gets the ant to deliver it there. Then comes the most iconic moment. The ant finds the underside of a leaf, and it bites down, hard. Its mandibles lock into the main vein of the leaf in what scientists call the death grip. The ant clamps down with such force that even after it dies, the jaws remain locked. The body hangs there, suspended, secured. This isn't random. The fungus has guided the ant to this exact position because it's the perfect launching pad for what comes next. Within a day or two, the ant dies, but the fungus is just beginning its work. Over the next couple of weeks, it digests the ant's insides, consuming everything except the exoskeleton, which it preserves as a shell, a protective container. And then, slowly, a structure begins to emerge from the back of the ant's head, a stalk. It grows upward, extending several centimeters, sometimes even longer. At the tip, a bulbous capsule forms, swelling with spores. Millions of them. When the conditions are right, when the humidity peaks in the early morning, this capsule ruptures. Spores rain down onto the forest floor below, where other ants are likely to walk. The cycle begins again. One ant infected and sacrificed can release enough spores to potentially infect dozens more. It's brutally efficient. Now, let's think about what had to evolve for this to work. The fungus needed to develop the ability to penetrate the ant's exoskeleton. It needed to avoid triggering an immune response strong enough to kill it before it established itself. It needed to produce chemicals that could manipulate the ant's nervous system, compelling it to leave the safety of the colony. It needed to guide the ant to a specific microclimate. It needed to induce the death grip at exactly the right moment. And it needed to preserve the body long enough to fruit and release spores. Every step of this process is coordinated. Every step is necessary. Miss one, and the entire strategy fails. And here's the truly mind-bending part. There are hundreds of species of Ophiocordyceps, and each one is specialized for a specific host. There's one that infects carpenter ants, another that infects bullet ants. Others target different ant species, or flies, or beetles, or moths. Each fungal species has evolved its own manipulation strategy, tailored to the behavior and biology of its particular host. They've even evolved different fruiting body structures, different spore release mechanisms, different chemical cocktails to control their hosts. Scientists have studied the compounds these fungi produce, and they've found alkaloids, complex organic molecules that interfere with neurotransmission. They've found ergot-like substances, similar to the compounds that cause hallucinations in humans. They've found proteins that seem to target specific neural pathways, but we still don't fully understand the complete mechanism. We know the fungus is manipulating the ant's brain. We just don't know all the details of how. One hypothesis is that the fungus doesn't directly control the brain at all. Instead, it might invade the muscles, forming a living network of hyphae that acts like a secondary muscular system. The fungus could be pulling the strings, literally contracting and relaxing muscles to make the ant walk, climb, and bite, even as the ant's own nervous system is failing. Some recent research supports this idea. When scientists examined infected ants, they found that the fungal cells were densely packed around muscle fibers, but relatively sparse in the brain itself. The ant might still be conscious, in some sense, but unable to control its own body, a prisoner in its own exoskeleton. Imagine that for a moment. Imagine knowing something is wrong, feeling your body moving without your permission, walking away from everything familiar, climbing to a place you don't want to be, and then locking your jaws onto a leaf, unable to let go, as something grows inside you. It's the stuff of nightmares. But for the ant, There's no awareness of the horror, no existential dread, just the chemical imperative, the hijacked signals that say climb, descend, bite, hold. And this has been happening for millions of years. The fossil record shows that these zombie ant fungi existed in the Eocene epoch nearly 50 million years ago. That means this relationship, this macabre dance between parasite and host, is ancient. It survived mass extinctions, climate shifts, the rise and fall of countless species. It's woven into the fabric of tropical ecosystems, an invisible thread that connects soil and leaf litter to the canopy high above. In some forests, infected ants are common enough that you can find them if you know where to look. Walk through the understory in the early morning, examine the undersides of leaves at that magic height of 25 centimeters, and you might spot them. Dead ants, jaws locked, fungal stalks emerging from their heads like grotesque antennae. And if you find one, you're looking at the end of a story that began weeks earlier, when a single spore landed on an unsuspecting ant and began its slow, methodical takeover. But here's something that might change how you think about this. From the fungus's perspective, this isn't evil. It's not cruel. It's just survival. The fungus has no consciousness, no malice, no awareness that it's causing suffering. It's following a script written by evolution, doing what it's been shaped to do over millions of years. And in the process, it plays a role in the ecosystem. It controls ant populations, preventing any one colony from becoming too dominant. It recycles nutrients, breaking down insect bodies and returning them to the soil. It provides food for other organisms that have learned to eat the fruiting bodies without being infected themselves. Nature isn't good or evil, it just is, and the zombie-making fungi are a perfect example of that moral neutrality. They're horrifying and beautiful at the same time. They're a testament to evolution's creativity, to the endless arms race between parasite and host, to the fact that life will find a way, no matter how strange, no matter how unsettling. So the next time you see a mushroom growing from something unexpected, pause for a moment. Consider the possibility that you're looking at the end of a story of control, of chemical manipulation, of one organism bending another to its will. And remember that in nature, the line between life and death, between self and other, is far more blurred than we like to think. Now, let's turn from the forests to the water, where another group of parasites has perfected an entirely different kind of control, one that ends not on a leaf, but in the depths. Picture a cricket on a warm summer evening. It's doing what crickets do, chirping, hopping through grass, searching for food and mates, staying alert for predators. The last thing on its mind, if we can call it a mind, is water. Crickets are terrestrial insects. They avoid large bodies of water because they can't swim. Water means drowning. Water means death. This fear is hardwired, shaped by millions of years of evolution. And yet, on this particular evening, this cricket is going to walk directly into a stream and drown itself, not by accident. Not because it slipped, but because something inside it is screaming, demanding, compelling it toward water with an urgency that overrides every survival instinct it possesses. Welcome to the world of hairworms, also known as Gordian worms. These are parasites that belong to the phylum Nematomorpha, and they've perfected one of nature's most dramatic examples of behavioral manipulation. Their life cycle requires water. They need to return to streams, ponds, or puddles to mate and lay eggs. But they spend most of their lives inside terrestrial insects like crickets, grasshoppers, and beetles. So they face a problem. How do you get a land-dwelling host to deliver you to water? The answer is both simple and terrifying. You make them want to. Let's follow the journey. It begins with an egg laid in water months or even years earlier. When the egg hatches, a microscopic larva emerges. It's tiny, barely visible, drifting in the current or sitting on aquatic vegetation. It waits. And eventually, if it's lucky, it gets consumed by a small aquatic insect, maybe a mayfly nymph or a mosquito larva. The hairworm larva burrows into this first host and forms a cyst, a protective capsule where it remains dormant. Now comes the critical step. That infected aquatic insect needs to be eaten by a terrestrial insect. This happens more often than you might think. Dragonflies, crickets, beetles. Many terrestrial insects supplement their diet with smaller aquatic prey.
When the cricket eats the infected mayfly, the hairworm cyst is transferred. Inside the cricket's gut, the cyst breaks open.
The hairworm larva emerges and begins to grow and grow and grow. Over the next several weeks or months, the hairworm develops inside the cricket's body cavity. It coils around organs, absorbs nutrients, and extends itself until it's sometimes 3 or 4 times longer than the host itself. Imagine that. A worm the length of your entire body coiled up inside your abdomen. How it fits is almost incomprehensible. The cricket's body becomes a living container, stretched and packed with parasite, and yet, somehow, the cricket survives. It continues eating, moving, functioning, even as this alien presence grows inside it. But the hairworm can't complete its life cycle inside the cricket. It needs to get back to water, and this is where the manipulation begins. As the hairworm approaches maturity, it starts producing proteins and other molecules that alter the cricket's nervous system. We don't fully understand all the mechanisms yet, but researchers have identified several neurotransmitters that are affected. One of the most important seems to be serotonin, that same chemical we talked about earlier. Infected crickets show significantly altered serotonin levels in their brains. Serotonin is involved in many behaviors, including mood regulation, aggression, and locomotion. But in insects, it also plays a role in phototaxis, the response to light. Normally, crickets are negatively phototactic at certain times. They avoid bright light and prefer darker, sheltered areas. This keeps them safe from predators. But when hairworm infection progresses, something changes. The cricket becomes attracted to light, especially the kind of light that reflects off water surfaces. Moonlight on a stream, starlight on a pond, the glimmer of wet stones. The cricket begins to wander. It leaves its normal habitat, moving with unusual determination toward any light source. And often, that light source is near water. The manipulation isn't random. The hairworm has somehow tuned the cricket's response to specifically seek out the environmental cues associated with water. Some studies suggest the parasite might even make the cricket more sensitive to humidity or the sound of running water. Every sense is hijacked, redirected toward a single goal. And then the moment arrives. The cricket reaches the edge of a stream or pond, and without hesitation, it jumps in. For a terrestrial insect, this is suicide. Crickets can't swim. Their legs thrash uselessly. Water fills their spiracles, the tiny breathing holes along their abdomen. They begin to drown. But just as the cricket hits the water, the hairworm makes its move. It bursts out, usually through a weak point in the exoskeleton, often near the joints or the abdomen. The emergence can take minutes or hours, as the long, thin worm uncoils itself and wriggles free. Watching this happen is surreal. The worm just keeps coming, meter after meter of dark, thread-like body emerging from an insect a fraction of its length. It's like a magician's trick, except it's real, and it's happening because of millions of years of evolutionary refinement. Once free, the hairworm swims away, finally in its element. It will find a mate, reproduce, and release eggs into the water. The cycle begins again, and the cricket, it usually dies, exhausted and drowned, its purpose fulfilled. But here's where this story gets even more interesting. This manipulation doesn't just affect one cricket. It affects ecosystems. In some streams, infected crickets represent a significant input of energy and nutrients. When they jump in and drown, they become food for fish and other aquatic predators. Scientists have calculated that in certain seasons, up to 60% of the diet of some trout species comes from terrestrial insects, many of them driven to water by parasites. The hairworms are essentially transferring energy from land to water, creating a bridge between two ecosystems that wouldn't otherwise be connected. Think about that ripple effect. A parasite infects a cricket. The cricket jumps into a stream. Fish eat the cricket and grow larger. Birds eat the fish. Nutrients cycle through the food web in ways that wouldn't happen without the parasite. Remove the hairworms, and you change the entire structure of these ecosystems. The parasites aren't just passengers. They're architects, shaping the flow of energy and matter through nature. And hairworms aren't the only parasites that drive hosts to water. There are flukes that infect snails and make them more likely to be eaten by birds near water. There are barnacles that alter crab behavior, making infected crabs migrate to estuaries where the barnacle's larvae have better survival chances. Water, it seems, is a common destination for parasites, and they've evolved countless ways to get their hosts there. The mechanism behind this water-seeking behavior is still being researched. Some scientists believe the parasite directly interferes with the central nervous system, altering neural circuits that control navigation and motor behavior. Others suggest the parasite might create a sensation of extreme thirst or discomfort that only proximity to water relieves. It's possible the cricket feels a need, an itch it can't scratch, a drive it can't understand, pulling it towards something it would normally fear. From a philosophical perspective, this raises profound questions. Where is the boundary between the cricket's will and the parasite's control? Does the cricket experience this as a choice or as a compulsion? Does it suffer, knowing something is wrong but unable to stop itself? Or does the manipulation happen at such a deep level that the cricket simply feels like it wants to go to the water, experiencing the hijacked desire as its own authentic preference? Is less than #0. 7# is greater than. We can't know what it's like to be an infected cricket. We can't access that subjective experience. But we can observe the behavior, measure the chemicals, map the neural changes, and what we find is always the same. A precise, targeted manipulation that serves the parasite's needs with ruthless efficiency. The cricket becomes a vehicle, a delivery system, a biological drone programmed with a single mission that it will complete even at the cost of its own life. And here's the unsettling part. This kind of manipulation isn't rare. It's everywhere. In forests, in deserts, in grasslands and wetlands. Thousands of parasite species have evolved similar strategies, each one fine-tuned to its particular host and environment. The natural world is full of organisms being steered by invisible hands, guided toward outcomes that benefit something other than themselves. So the next time you see an insect behaving strangely, moving with unusual purpose towards something that seems dangerous, consider the possibility that it's not alone in its body, that it's carrying a passenger with its own agenda. And that what you're witnessing isn't madness or confusion, but control, exerted from the inside out, one neuron at a time. Now, let's move from behavioral manipulation to something even more dramatic. Parasites that don't just change how their hosts act, but physically transform what their hosts are. Imagine waking up one morning and discovering that your body has been remodeled overnight. Your shape has changed, your internal organs have been rearranged, your reproductive system no longer belongs to you. And yet, you go about your day as if everything is normal, caring for offspring that aren't yours, nurturing a future that will never be your own. This isn't science fiction. This is what happens when certain parasites don't just manipulate behavior, but restructure the host's entire body to serve their needs. Let's travel to the ocean, to the rocky shores and coral reefs where crabs scuttle across the bottom, filtering food, defending territory, searching for mates. Now, focus on one particular crab. It looks normal from the outside. Strong claws, hard shell, moving with that characteristic sideways gait. But inside, something has taken over so completely that the crab is no longer really a crab. It's a factory, a vehicle, a mobile life support system for a parasite that has rewritten its biology from the inside out. The parasite is called Sacculina. It's a type of barnacle, though it looks nothing like the barnacles you might have seen on boat hulls or pier pilings. Sacculina has evolved beyond recognition, becoming something so specialized, so intimately adapted to its parasitic lifestyle, that scientists initially didn't even realize it was a barnacle. It has no shell, no legs, no digestive system. It's essentially a living root system wrapped around the crab's internal organs with a small external sac for reproduction. And the way it accomplishes this transformation is both elegant and horrifying. It begins with a larva. Female Sacculina larvae swim through the ocean until they detect chemical signals from a crab. When they find a suitable host, they settle on its shell and begin their invasion. But they don't burrow through the hard exoskeleton like you might expect. Instead, they find a soft spot, usually near the base of a leg or at a joint. The larva injects a small mass of cells through this opening. Just cells. No organs, no structure, just the raw biological material needed to start an infection. These cells begin to grow, spreading throughout the crab's body like roots through soil. They extend tendrils that wrap around the crab's gut, its nerve centers, its reproductive organs. The parasite taps into the crab's circulatory system, siphoning off nutrients directly from the bloodstream. Within weeks, Sacculina has colonized nearly every part of the crab's interior. If you could see through the shell, you'd find the parasite everywhere, an alien presence so thoroughly integrated that removing it would kill the host. But Sacculina doesn't just steal resources, it transforms the crab. And this is where things become truly remarkable. If the host is a female crab, Sacculina castrates it chemically, shutting down its reproductive system. The crab will never produce eggs of its own again, its ovaries atrophy, its reproductive hormones are suppressed. But here's the twist. The crab still exhibits all the maternal behaviors it would normally use to care for its own eggs. It still cleans its abdomen, still fans water over the egg mass, still protects it from predators. Except now, the eggs it's caring for belong to Sacculina. The parasite eventually produces an external reproductive sac that emerges from the crab's abdomen, right where the crab's own eggs would normally be attached.
To the crab's nervous system, this sac feels like its own egg mass.
The manipulation is so complete that the crab's maternal instincts are hijacked, redirected entirely to the parasite's benefit. The crab becomes a surrogate parent, devoting all its energy and protective behavior to ensuring the survival of its parasite's offspring. But what about male crabs? This is where Sakhalina's manipulation becomes even more extraordinary. When the parasite infects a male crab, it doesn't just castrate it, it feminizes it. The male's body changes shape. Its abdomen broadens, becoming more like a female's. Its claws shrink. Its behavior shifts. The infected male begins acting like a female, even though it's genetically and anatomically male. And just like infected females, it cares for Sakhalina's reproductive sac, as if it were its own eggs, fanning water, cleaning, protecting. How does the parasite do this? By manipulating hormones. Crustaceans, like many animals, have hormones that control sexual development and behavior. In crabs, these include ecdysteroids and juvenile hormone-like compounds. Sakhalina produces its own versions of these hormones, or blocks the crab's natural production, essentially rewriting the hormonal blueprint that determines what the crab's body looks like and how it behaves. The result is a creature that's been biologically reprogrammed to serve a different master. And the crab never reproduces again. Its entire reproductive future has been erased. From an evolutionary perspective, this is death. It doesn't matter if the crab's body continues living for months or years. Its genes won't be passed on. Its lineage ends. It's become what biologists call a zombie, not in the Walking Dead sense, but in the sense that it's biologically dead while still being metabolically alive. It exists only to serve the parasite. Now, you might think the crab would resist, that its immune system would fight back, that evolution would have prepared it for this kind of invasion. And crabs do have immune systems. They can recognize foreign cells and mount responses against bacteria, viruses, and other pathogens. But Sakhalina has evolved ways around these defenses. Its cells mimic the crab's own cells closely enough that the immune system doesn't recognize them as foreign. And by the time the infection is established, it's too late. The parasite has become so integrated with the host's tissues that attacking it would mean attacking the host's own body. This kind of parasitism is called parasitic castration, and it's more common than you might think. It's been documented in snails, fish, insects, and various marine invertebrates. The strategy makes evolutionary sense. Why kill your host quickly when you can keep it alive and turn its entire body into a resource for your own reproduction? From the parasite's perspective, the host is real estate, energy storage, transportation, and childcare, all rolled into one. But let's think about what this means for the crab. Does it know something is wrong? Does it feel the loss of its reproductive capability? Does it experience confusion when its body changes shape? We can't know. Crabs don't have the kind of consciousness we do. Their nervous systems are simpler, their awareness more basic. But they do have behaviors, preferences, drives, and all of those have been altered, redirected, stolen by an organism that's turned them into an involuntary participant in someone else's life cycle. There are other examples of body-snatching parasites. There's a fluke called leucochloridium that infects snails and produces colorful, pulsating structures in the snail's eye stalk. These structures look like caterpillars, which attracts birds. The birds eat the snail, and the parasite completes its life cycle in the bird's gut. The snail's entire body becomes a lure, a visual advertisement designed to get it eaten. its eyes are transformed into bait. Or consider parasitic wasps that lay eggs inside caterpillars. When the wasp larvae are ready to pupate, they chew their way out of the caterpillar's body. You'd think the caterpillar would be dead or at least incapacitated, but no. It survives, and it spins silk over the wasp cocoons, protecting them from predators and weather. The caterpillar has become a bodyguard, defending the very creatures that ate it from the inside. And when the wasps emerge as adults and fly away, the caterpillar finally dies, its purpose fulfilled. These transformations reveal something profound about biology. The boundary between organisms isn't as clear as we like to think. Your body feels like a unified whole, a single entity with clear edges, but you're actually an ecosystem. Trillions of bacteria live in your gut, on your skin, in your mouth. Mitochondria in your cells have their own DNA, remnants of ancient bacteria that merged with our ancestors billions of years ago. Viruses have inserted their genetic material into your genome. Pieces of you aren't entirely you. And in the case of parasitized crabs, the question of where the crab ends and the parasite begins becomes impossible to answer. The crab's body is still crab. Its DNA is still crab DNA. But its behavior, its hormones, its reproductive future, all of that belongs to Sakulina now. The crab is a collaboration, a hybrid entity serving two genomes, though only one of them benefits. It's a living reminder that control doesn't always require a brain hijacking neurons. Sometimes control means rewriting the body itself, turning flesh and instinct into tools for a different kind of survival. So, when you think about identity, about what makes you you, remember the parasitized crab. Remember that bodies can be occupied, transformed, redirected, and that in nature the self is always negotiable, always vulnerable to forces that see you not as an individual, but as an opportunity. Next, we'll explore the chemistry in even greater detail, diving into the molecular weapons parasites use to dismantle defenses and rewrite the rules of biology itself. Your body is having a conversation right now. Trillions of messages are being sent every second, cells talking to each other, organs coordinating, your immune system scanning for threats, your brain processing sensations, and all of this communication happens through chemistry. Molecules carrying information, binding to receptors, triggering responses. It's a language so precise that changing a single molecule, adding or removing one atom, can completely alter the message. And parasites have learned to speak this language fluently. They've become master chemists, crafting molecular weapons that can disarm defenses, rewrite instructions, and turn the host's own biology against itself. Let's start with the most fundamental challenge any parasite faces when it enters a host: the immune system. Your immune system is extraordinary. It can recognize millions of different foreign molecules, distinguish self from non-self, and mount targeted attacks against invaders. White blood cells patrol your bloodstream like security guards, checking identification, looking for anything that doesn't belong. If they find a bacterium, a virus, or a parasite, they attack. They engulf it, they tag it for destruction. They release toxic chemicals to kill it. They remember it, so if it ever comes back, the response is even faster and more overwhelming. For a parasite, this is a problem. How do you survive in an environment that's actively trying to kill you? The answer is chemical warfare. Parasites produce molecules that interfere with immune signaling. They mimic host molecules to avoid detection. They suppress inflammation. They redirect immune responses. And they do all of this with a sophistication that rivals anything human medicine has developed. Take the parasite schistosoma, a flatworm that causes a disease called schistosomiasis, affecting over 200 million people worldwide. When schistosoma enters your body, usually through skin contact with contaminated water, your immune system immediately recognizes it as foreign. Immune cells rush to the site. They try to wall it off, to kill it before it can establish itself, but schistosoma has a trick. It coats itself with molecules stolen from your own body. It covers its surface with your proteins, your carbohydrates, your lipids. To your immune system, it starts to look like self. The molecular disguise is so effective that the parasite can live in your bloodstream for years, sometimes decades, right under the nose of an immune system that would normally destroy it within hours. But camouflage is just one strategy. Other parasites take a more aggressive approach. They actively suppress the immune system, releasing molecules that interfere with the signaling pathways immune cells use to communicate. Remember, your immune response is coordinated through chemical messengers called cytokines. These are proteins that tell immune cells where to go, what to do, how strongly to respond. If a parasite can block cytokine production or prevent cytokines from binding to their receptors, it can essentially silence the alarm bells that would normally trigger an immune attack. Toxoplasma gondii, that mind-controlling parasite we talked about earlier, does exactly this. It produces proteins that interfere with the host's production of inflammatory cytokines. It also manipulates a cellular pathway called NF-kappa B, which is like a master switch for immune responses. By tweaking this pathway, Toxoplasma can turn down inflammation just enough to survive without completely disabling the immune system, which would leave the host vulnerable to other infections and potentially kill it. The balance is delicate. The parasite needs the host alive and functional, but not so vigilant that it gets destroyed. Now, let's zoom in even further to the molecular level, where parasites are conducting chemical operations that would impress any pharmaceutical company. Consider neurotransmitters again. We talked about how parasites manipulate these molecules to change behavior. But let's look at the specific chemistry involved. Neurotransmitters are small, organic molecules, usually built from amino acids. Dopamine, for example, is synthesized from the amino acid tyrosine through a series of enzymatic reactions. Each step requires a specific enzyme to catalyze the transformation. If you want to increase dopamine levels in the brain, you could introduce more tyrosine, or you could increase the activity of the enzymes that convert tyrosine to dopamine. Or you could block the enzymes that break down dopamine after it's been released. Toxoplasma does the second option. It carries genes for tyrosine hydroxylase, the rate-limiting enzyme in dopamine synthesis. When the parasite infects brain tissue, it expresses this enzyme, effectively setting up its own dopamine production facility inside the host neurons. The result is a local increase in dopamine concentration, which alters neural activity in nearby circuits. The host's brain is still making its own dopamine through normal pathways, but now there's an additional source, a rogue factory pumping out extra neurotransmitter in specific locations. And because neurotransmitters work at very low concentrations, even a small increase can have profound effects on behavior. Other parasites work differently. Some produce molecules that mimic neurotransmitters closely enough to bind to the same receptors. Imagine your brain's receptors as locks and neurotransmitters as keys. Each receptor only opens for a specific molecular shape. But if a parasite can produce a molecule that's similar enough, it can slip into that lock and trigger the same response, or sometimes block the lock entirely. preventing the real neurotransmitter from binding. This is called molecular mimicry, and it's incredibly common in parasitic infections. Then there's GABA, gamma-aminobutyric acid, the brain's primary inhibitory neurotransmitter. GABA is what calms neural activity, reduces anxiety, promotes relaxation. It's also involved in motor control and various cognitive functions. Some parasites produce GABA-like molecules or enzymes that increase GABA levels in the host's brain. The effect can be sedation, reduced motor coordination, or altered decision-making. For a parasite that needs its host to be less reactive, less likely to groom itself and remove the parasite, or less aggressive toward the parasite's reproductive stages, boosting GABA makes perfect sense. And it's not just neurotransmitters. Parasites also manipulate hormones, those long-range chemical messengers that coordinate physiology across the entire body. We saw this with Saculina feminizing male crabs. The mechanism involves molecules that either mimic or block sex hormones. In crustaceans, hormones like methylpharmacoate and ecdystroids control molting, reproduction, and sexual differentiation. By producing analogues of these hormones, or by blocking their receptors, parasites can essentially reprogram the host's endocrine system, the network of glands and hormones that regulates growth, metabolism, and reproduction. The precision here is astounding. These aren't crude poisons that simply damage tissue. These are targeted molecular interventions that alter specific pathways while leaving others intact. It's like hacking into a computer and changing a few lines of code. The system still runs, it still performs most of its functions, but the output is different, the behavior is different, and the parasite benefits. Now, think about what this means from an evolutionary perspective. For a parasite to evolve these capabilities, it needs to acquire or develop genes that code for the right enzymes, the right structural proteins, the right signaling molecules. This takes time, countless generations of trial and error, natural selection, favoring parasites that happen to produce molecules that slightly improve survival or reproduction. Over millions of years, these small advantages accumulate. The parasites become better chemists, more precise, more effective. And the hosts aren't passive. They evolve too. They develop new immune mechanisms. They change receptor structures so that parasitic molecules no longer bind as effectively. They increase the diversity of their immune molecules, making it harder for parasites to evade detection. This is the evolutionary arms race we mentioned earlier, played out in chemical terms. Each side evolves countermeasures. Each side develops new weapons. And the result is an escalating conflict where neither side can ever fully win, because as soon as one gains an advantage, selection pressure intensifies on the other. In your own body, right now, this chemical conversation is ongoing. Your gut bacteria are producing metabolites that influence your brain chemistry, your mood, your digestion. Your immune cells are releasing cytokines in response to minor infections you're not even aware of. Your neurons are constantly adjusting neurotransmitter levels based on what you're thinking, feeling, experiencing. You are, in a very real sense, a chemical system. And like any system, you can be influenced by external inputs, by molecules that don't originate from your own genome, but that speak the language your cells understand. This is why studying parasites is so valuable. They've spent eons learning how to manipulate biology at the chemical level. And by understanding their techniques, scientists can learn how to do similar things, but for therapeutic purposes. Want to treat depression? Look at how parasites increase dopamine. Want to reduce inflammation?
Study how parasites suppress immune responses. Want to control pain? Examine the molecules parasites use to numb host tissues. Nature has already run billions of experiments. Parasites are the results of those experiments, and hidden in their chemistry are potential solutions to human medical challenges. But there's also something humbling about this. The realization that consciousness, personality, decision-making, all the things we think of as uniquely ours, fundamentally ourself, are built on chemistry. And chemistry can be altered. Parasites prove this every day. They prove that who you are is inseparable from the molecules flowing through your brain. Change the molecules, and you change the person. It's not magic, it's not mysterious. It's just biology, operating according to laws we're only beginning to fully understand. So as you lie there tonight, drifting towards sleep, your own brain is conducting its chemical symphony, adenosine building up, making you drowsy, melatonin rising, telling your body it's time to rest, GABA increasing, quieting neural activity. It's all chemistry. And it's all beautiful. A coordinated dance of molecules that, when everything works correctly, allows you to let go, to surrender control, and to drift into unconsciousness. Next, we'll explore parasites with the most complex life cycles imaginable, organisms that must pass through multiple hosts, each one serving a different purpose, each transition requiring perfect timing and precise manipulation. Imagine trying to travel from one side of the world to the other, but you can't fly, you can't swim across oceans, and you can't walk more than a few meters on your own. You're microscopic, you're fragile, you're completely dependent on other organisms for transportation. Now imagine that your journey requires not just one vehicle, but 3 or 4 different ones, each operating in completely different environments. You need to be eaten by the right animal, at the right time, in the right place. Miss any step, and your lineage ends. This is the reality for parasites with complex life cycles. And somehow, against astronomical odds, they make it work. Let's follow one of the most remarkable journeys in all of parasitology, the story of Toxoplasma gondii, that same parasite we've encountered before, but now we'll see its full life cycle, the complete odyssey it must undertake to survive. This journey involves 3 types of hosts, spans multiple ecosystems, and requires a level of biological coordination that seems almost impossible. Yet, it's been happening successfully for millions of years. The journey begins in a cat. Not just any cat, but the intestines of a cat, because Toxoplasma can only complete sexual reproduction inside feline digestive systems. No one knows exactly why evolution locked this parasite into such a specific requirement, but that's where we are. Inside the cat's intestines, male and female Toxoplasma cells meet, fuse, and produce oocysts, tough-walled structures that contain the parasite's offspring. These oocysts are shed in the cat's feces, millions of them, released into the environment where they can survive for months or even years in soil and water. Now the waiting begins. The oocysts need to be consumed by an intermediate host. This could be a mouse, a rat, a bird, a pig, a sheep, or even a human. Toxoplasma isn't picky at this stage. Any warm-blooded animal will do. The oocysts contaminate vegetation, water sources, soil. They're virtually invisible, and they're incredibly resilient, able to withstand freezing, drying, and exposure to sunlight. They're biological time capsules, waiting for the right moment. Is less than #0. 7# is greater than. Let's say a mouse drinks from a puddle contaminated with oocysts. The mouse swallows them without knowing. In the mouse's stomach, the oocysts break open, releasing sporozoites, the active, infectious form of the parasite. These sporozoites invade the intestinal lining, enter the bloodstream, and begin to spread throughout the body. They're looking for specific tissues, brain, muscle, heart, places where they can hide from the immune system and establish long-term infections. Once they reach these tissues, the sporozoites invade cells and transform into a different form called tachyzoites. These are the rapidly dividing form, the expansion phase. Tachyzoites multiply quickly, bursting out of cells and invading new ones, spreading the infection throughout the host. The mouse's immune system responds, but as we discussed earlier, Toxoplasma has ways of evading and suppressing immunity. Eventually, the acute infection transitions to a chronic phase. The tachyzoites slow down, form those protective cysts we talked about, and enter a dormant state called bradyzoites. These cysts can remain in the mouse's brain and muscles for the rest of its life, metabolically quiet, waiting. But here's the key. Toxoplasma can't complete its life cycle in the mouse. It needs to get back to a cat. And this is where that behavioral manipulation comes in. The loss of fear, the attraction to cat urine. The infected mouse becomes more likely to be caught and eaten. And when a cat consumes an infected mouse, the tissue cysts in the mouse's flesh are freed in the cat's digestive system. The bradyzoites emerge, transform again, and now they can undergo sexual reproduction. The cycle completes. New oocysts are produced and shed into the environment. The journey begins again. Think about the precision required here. The parasite has to exist in multiple forms, each adapted to a different environment and host. It has to time its transitions perfectly. It has to manipulate host behavior at exactly the right stage. It has to evade immune responses in multiple different species, each with its own unique immune system. And it has to accomplish all of this without any brain, without any central coordination, just following genetic programming that's been refined over millions of years. Now, Toxoplasma's life cycle, as complex as it is actually relatively simple compared to some parasites. Consider the lancet liver fluke, Dicrocoelium dendriticum. This parasite requires 3 hosts to complete its life cycle, and each transition involves a different manipulation strategy. It starts in a snail. Fluke eggs are eaten by snails grazing on grass. Inside the snail, the eggs hatch and develop into a larval stage. But the snail's immune system responds by producing mucus to wall off the infection. The fluke larvae end up concentrated in balls of slime, which the snail eventually expels, coughing them out onto vegetation. This is where it gets interesting. Ants are attracted to this slime. They eat it, consuming hundreds of fluke larvae in the process. The larvae migrate through the ant's body, and most of them settle in the ant's abdomen, forming cysts. But 1 or 2 larvae take a different path. They travel to the ant's brain, specifically to the subesophageal ganglion, a cluster of nerve cells that controls the ant's mandibles, its jaws. And at night, when temperatures drop, these brain-dwelling larvae cause the ant to do something bizarre. The ant climbs to the top of a grass blade, clamps its jaws around the blade, and stays there, locked in place. This behavior is called the summit disease, and it's remarkably consistent across infected ants. They climb, they bite, they hold on, and there they remain, exposed and vulnerable, until a grazing animal like a sheep or cow comes along in the morning and eats the grass blade, ant and all. Inside the grazing animal's digestive system, the fluke larvae are finally freed. They migrate to the liver and bile ducts, where they mature into adult flukes, reproduce, and release eggs that pass out in the animal's feces. Those eggs contaminate the grass. Snails eat them. The cycle starts over. 3 hosts, 3 different manipulation strategies. Snails are made to expel slime balls. Ants are made to climb and bite. Grazing animals are simply used as the final reproductive host, though some research suggests the flukes may even influence grazing behavior, making infected animals more likely to eat low grass where infected ants are waiting. Every step of this cycle is necessary. Skip one, and the entire lineage collapses. Yet somehow, enough flukes make it through each stage to keep the species thriving. And there are even more complex cycles. Some parasites use 4 or 5 hosts. Some have free living stages that must survive in harsh environments between hosts. Some produce different larval forms for aquatic and terrestrial hosts. The diversity is staggering, but the underlying principle is the same. These parasites have evolved to exploit the food webs and ecological relationships that connect different species. They're essentially hitchhiking through ecosystems, using predator-prey relationships as transportation networks. From an evolutionary perspective, how does something this complex even evolve? It must have started simpler. Perhaps an ancestral toxoplasma only needed one host. Then, at some point, a genetic mutation allowed it to survive briefly in a second host. Any parasite that could use that second host as a bridge to reach more of the first host would have had an advantage. Over time, the 2-host cycle became obligatory. Then, maybe a third host was added, further improving transmission efficiency. Each step had to provide some benefit, some increase in reproductive success, or it wouldn't have been maintained by natural selection. The result is what we see today. Parasites with life cycles so intricate they resemble elaborate machines, with each host serving as a different component. And the coordination required is encoded entirely in the parasite's genome, in the proteins it produces, in the chemicals it releases at each stage. There's no conscious planning, no foresight, just the accumulated wisdom of millions of years of trial and error written in DNA. And here's something that might shift your perspective. These complex life cycles aren't failures or inefficiencies, they're adaptations. Having multiple hosts provides redundancy. If one host population declines, the parasite might still persist through other routes. Multiple hosts also allow parasites to exploit different ecological niches, accessing resources and reproductive opportunities that wouldn't be available with a simpler life cycle. It's costly, yes. Most individual parasites die without completing the journey, but for the lineage as a whole, the strategy works. The genetic instructions for this complexity continue to be passed down generation after generation. So when you think about your own life, your own journey from birth through development and into adulthood, consider that you're following a kind of life cycle too. Not as complex as a parasite's perhaps, but still a coordinated sequence of stages, each with its own challenges and requirements. And just like parasites, you're navigating an environment full of other organisms, forming relationships, depending on others for survival, contributing to cycles larger than yourself. We're all participants in ecosystems we barely understand, playing roles in stories that began long before we arrived and will continue long after we're gone. Next, we'll look at parasites that don't just manipulate hosts, but make them build structures, transforming their bodies into architects,
creating shelters and fortresses for the parasites growing inside them. Take a walk through any forest or garden, and you'll notice strange growths on plants, bulbous swellings on stems, spherical balls attached to leaves, spiky protrusions on twigs. They look like fruit, like seed pods, like natural parts of the plant. But they're not. They're galls, and inside each one is a parasite that has done something extraordinary. It has convinced the plant to build it a house, a custom-designed shelter with thick walls, controlled climate, and even food production built right in. The plant provides everything, willingly constructing a nursery for an organism that's stealing its resources. And the parasite accomplishes this without a brain, without hands, without any apparent consciousness, just chemistry and timing. Let's start with the basics. A gall is an abnormal growth of plant tissue triggered by the presence of another organism, usually an insect or mite. But this isn't like a tumor, a random mass of uncontrolled cells. Galls are highly structured. They have specific shapes, specific internal architectures, specific chemical compositions. Each species of gall-making parasite produces its own distinctive gall, so consistent in appearance that botanists and entomologists can identify the parasite species just by looking at the gall it creates. This isn't accident. This is biological engineering. Here's how it works. A female gall wasp, for example, lands on an oak tree. She has a needle-like ovipositor, a specialized egg-laying organ, and she uses it to inject an egg into the plant tissue, usually into a bud or young leaf. But she doesn't just deposit the egg and leave. She also injects a cocktail of chemicals, proteins, and possibly even viruses or bacteria that live symbiotically with the wasp. These chemicals are the instructions. They're molecular blueprints that tell the plant cells what to build. The plant cells respond. They begin to divide rapidly, but not randomly. They form layers, an outer protective shell, often thick and woody, an inner nutritive layer, soft and rich in starches and proteins. Sometimes there are even specialized chambers, air pockets, drainage channels. The architecture varies depending on the species, but the principle is the same. The plant is building a structure that benefits the parasite, not itself, and the plant does this using its own energy, its own nutrients, its own cellular machinery. Why doesn't the plant just ignore the signal? Why does it cooperate with an organism that's clearly exploiting it? The answer lies in how plant cells respond to chemical signals. Plants use hormones to coordinate growth and development. Auxins promote cell division and elongation. Cytokinins stimulate cell division and differentiation. Gibberellins regulate stem growth. Ethylene controls fruit ripening and stress responses. These hormones work in carefully balanced concentrations, and changing those concentrations changes how the plant grows. The gall-making parasites have evolved to produce or cause the plant to produce abnormal levels of these hormones. They hijack the plant's developmental pathways. It's similar to what Saccharina does to crabs or what Toxoplasma does to mice, but instead of manipulating animal neurons, it's manipulating plant cells. The plant can't distinguish between legitimate growth signals and parasitic mimics. It follows the instructions because that's what its cells are programmed to do when they receive those chemical messages. Inside the gall, the wasp larva hatches and begins to feed. It doesn't eat the plant directly in the way a caterpillar would, chewing through leaves. Instead, it feeds on the nutritive tissue the plant has created specifically for it. This tissue is essentially baby food, Pre-digested nutrients in an easily accessible form. The larva grows, molts, develops. It's warm. It's protected from predators, from weather, from parasites of its own. The gall is a fortress and a buffet combined. And the diversity of galls is astonishing. On oak trees alone, there are hundreds of different gall-forming species, each producing a unique structure. Some galls look like perfect spheres. Others resemble spiky sea urchins. Some are smooth and green. Others are woody and brown. There are galls that look like flowers, galls that look like berries, galls that form intricate rosettes. And each design serves a purpose, evolved over millions of years to optimize the parasite's survival. For instance, some galls have thick walls that make it difficult for parasitoid wasps, which prey on gall makers, to drill through and reach the larva inside. Other galls produce toxic or bitter chemicals that deter herbivores from eating them. Some galls have spongy interiors that insulate the larva from temperature extremes. Others have hard, dense chambers that protect against physical damage. Its defensive architecture, designed through evolutionary trial and error, is less than #0. 7# is greater than, but it's not just insects that make galls. Mites, nematodes, fungi, and bacteria can also induce gall formation. Each uses slightly different chemical strategies, but the result is similar. The plant builds something it wouldn't normally build, something that serves the parasite's needs. And in some cases, multiple parasites will inhabit the same gall, creating a complex micro-ecosystem within a structure the size of a marble. Now, you might wonder whether galls harm the plant. Sometimes they do. Heavy gall infestations can stunt growth, reduce fruit production, or cause branches to die. But often the damage is minimal. The plant is large. It has excess resources. Losing a few leaves or branches to gall formation doesn't threaten its survival. In fact, some researchers suggest that plants might tolerate galls as a kind of compromise. It's metabolically cheaper to build a gall and contain the parasite than to mount a full immune response and kill it. The gall becomes a quarantine zone, keeping the infection localized. There are even cases where galls might benefit the plant indirectly. Some galls attract predators and parasitoids that attack other, more harmful insects. By hosting gall makers, the plant inadvertently recruits bodyguards. It's an unintended consequence, but evolution doesn't care about intentions. It only cares about outcomes. If harboring some galls helps the plant survive better overall, then tolerance will be selected for. Let's shift to another kind of construction behavior. Some parasites don't make their hosts build structures on their own bodies. Instead, they make hosts build structures in the environment. There's a parasitic wasp called Hymenoepimesis that targets orb weaver spiders. The female wasp stings a spider, temporarily paralyzing it, then lays an egg on its abdomen. The egg hatches, and the larva attaches to the spider, feeding on its bodily fluids through a small wound. The spider recovers and goes about its life, building webs, catching prey, seemingly unaffected. But when the wasp larva is ready to pupate, something changes. It injects chemicals into the spider, and those chemicals alter the spider's web-building behavior. Spiders normally build circular orb webs with a spiral of sticky silk to catch insects. But the parasitized spider builds a completely different structure. It creates a dense, reinforced web with thickened radial supports and no sticky spiral. This isn't a web for catching prey. It's a web designed to support a cocoon. the spider is building a scaffold for the wasp's pupation. Once the web is complete, the wasp larva kills the spider, consumes its remaining bodily fluids, and spins a cocoon attached to the reinforced web. The cocoon hangs safely above the ground, protected from ants and other ground-dwelling predators. The web structure distributes weight and resists wind and rain. It's a custom-engineered support system built unwittingly by the spider under the parasite's chemical influence. How does the wasp larva control such complex behavior? Spiders build webs through an intricate sequence of movements controlled by their central nervous system. Changing the web architecture means changing that behavioral sequence, reprogramming the spider's motor patterns. The chemicals the wasp injects likely target the spider's neurotransmitters or hormones, altering the neural circuits that govern web construction. It's precise, it's repeatable, and it's been documented across multiple wasp and spider species, suggesting this manipulation has evolved independently multiple times. These examples reveal something profound about the natural world. Behavior isn't just about individual action, it's about construction, about shaping the environment to suit needs. Humans build houses, cities, farms. We think of this as uniquely human, a product of intelligence and culture. But in reality, countless organisms modify their environments. Birds build nests, beavers build dams, termites build mounds, and parasites, microscopic and mindless, build too. They just use other organisms as their construction workers. The key difference is control. When you build something, you're executing your own plan, following your own goals. But when a plant builds a gall or a spider builds a wasp cocoon web, it's executing someone else's plan. The structure serves the parasite's goals, not the host's, and the host has no choice in the matter. Its biology has been hijacked, repurposed, turned into a tool for another organism's reproduction. This is architecture at its most fundamental level, not blueprints and bulldozers, but molecules and cells, chemicals that say grow here, divide there, build this shape, this thickness, this texture. And the fact that parasites have evolved to manipulate these processes shows just how malleable biology really is. Given the right signal, cells will build almost anything. They don't question. They don't resist. They simply follow the chemical instructions they receive. So, when you see a gall on a tree branch or a strange web pattern, pause for a moment. You're looking at collaboration, unwilling and unknowing, but collaboration nonetheless. Two organisms, one directing, one building, creating structures that neither could produce alone. It's a reminder that in nature, boundaries are permeable, that agency can be transferred, and that control is always a matter of chemistry and information flowing between living things. Next, we'll explore the darkest manipulation of all. Parasites that drive their hosts to the ultimate sacrifice, orchestrating deaths that serve no purpose except the parasite's continuation. Death is usually something organisms try to avoid. Every instinct, every evolved behavior, every physiological mechanism is designed to keep you alive long enough to reproduce and pass on your genes. Fear, pain, hunger, thirst. All of these are signals that guide you away from danger and toward survival. But what if those signals could be rewritten? What if an organism could be made to walk willingly toward its own death, not in confusion or accident, but with apparent purpose? This is the realm of suicide-inducing parasites, organisms that have evolved to make their hosts sacrifice themselves at precisely the moment when that death serves the parasite's reproduction. We've already met some of these parasites. The hairworms that drive crickets into water, the zombie ant fungi that guide ants to the perfect death grip. But let's look more closely at what suicide manipulation really means and why it represents the ultimate form of parasitic control. Because making a host die isn't impressive on its own. Any parasite can kill through tissue damage, immune dysfunction, or resource depletion. What's remarkable is making the host die in a specific way, at a specific time, in a specific place. That requires not just control, but coordination, timing, strategic thinking encoded in chemistry. Let's return to that cricket standing at the edge of a stream, driven by the hairworm growing inside it. The cricket's entire evolutionary history has programmed it to avoid water. Its ancestors that were careless around water drowned and didn't pass on their genes. The ones that survived were cautious, careful, water-averse. That trait has been reinforced over millions of generations. And yet, here stands this cricket, about to override millions of years of natural selection because a parasite needs to get wet. The moment the cricket jumps, it's committing suicide in the most literal sense. It can't swim. It will drown within minutes. But from the parasite's perspective, this death is perfectly timed. The hairworm is sexually mature. Its body has reached full size, coiled impossibly tight inside the cricket's abdomen. It's ready to emerge, to mate, to reproduce. But it can only do that in water, so the cricket's death isn't wasteful, it's necessary. It's the final service the host provides, a last act that ensures the parasite's genes continue into the next generation. And the cricket isn't aware of the sacrifice. It doesn't know it's being manipulated. It just feels compelled. The chemical changes in its brain create a drive, an irresistible urge that feels, to whatever extent a cricket can feel, like its own decision. The line between coercion and choice becomes meaningless when the choice itself is an illusion created by altered neurotransmitters. Now consider a different example. There's a species of flatworm called Leucochloridium paradoxum that infects amber snails. The life cycle is complex, involving both snails and birds. The parasite needs to get from the snail into a bird to complete its reproduction. But birds don't normally eat snails. Snails are cryptic, slow-moving, hiding in leaf litter and under logs. They're nearly invisible to predators, so the parasite has to make the snail visible. And it does this in the most visually striking way imaginable. The flatworm forms brightly colored, pulsating structures inside the snail's tentacles, those eye stalks that protrude from the snail's head. These structures are called brood sacs, and they're filled with parasite larvae, but they don't look like parasites. They look like caterpillars, fat, colorful, wriggling caterpillars, the kind that birds love to eat. The brood sacs pulse rhythmically, mimicking the movement of real caterpillars, and they're positioned right at the tips of the snail's eye stalks, in plain view. But that's not all. The infected snail's behavior changes. Normally, snails avoid light. They're most active at night or in shaded areas during the day, but infected snails seek out bright, exposed locations. They climb to the tops of grass blades, they position themselves in full sunlight, and they wave their infected eye stalks around, making the fake caterpillars even more visible. It's like putting up a neon sign that says, "Eat me." Eventually, a bird spots the snail. It pecks at what it thinks is a caterpillar. It bites off the snail's eye stalk, consuming the brood sac full of parasite larvae. The larvae survive in the bird's digestive system, mature into adult flatworms, reproduce, and release eggs that pass out in the bird's droppings. Those eggs contaminate vegetation. Snails eat the contaminated leaves. The cycle continues. And the snail? It often survives the loss of its eye stalk. Snails can regenerate, but the parasite can produce multiple brood sacs. So the process repeats, the snail offering up its body parts one at a time until it's finally killed or the infection runs its course. This is a delayed suicide. The snail doesn't die immediately, but its behavior ensures it will eventually be eaten. It's sacrificing pieces of itself, and ultimately its life, to fulfill the parasite's reproductive needs. And again, the snail has no awareness of this. It's not making a conscious choice. Its nervous system has been altered to seek light instead of shade, to climb instead of hide, to display instead of conceal. Let's talk about another form of orchestrated death. Some parasites manipulate hosts to make them more vulnerable to predation, not through behavior changes, but through physiological impairment. There's a protozoan parasite called toxoplasma that infects fish. Infected fish show reduced predator avoidance. They swim more slowly. They're less responsive to threats. They spend more time in open water where they're exposed to birds and larger fish. The impairment isn't random. It's strategic. The parasite needs the fish to be eaten by a bird to complete its life cycle, so it makes the fish easier to catch. Similarly, some parasitic worms that infect small fish cause them to swim erratically near the surface, behavior that attracts predatory birds. The fish essentially become targets, their normal, evasive maneuvers replaced by conspicuous, vulnerable swimming patterns. When a bird catches and eats the infected fish, the parasite moves into the bird's gut, matures, and reproduces. The fish's death was the necessary bridge. From an evolutionary standpoint, these suicide manipulations only make sense if they increase the parasite's transmission success more than enough to offset the cost of killing the host. And clearly, they do. These strategies have persisted for millions of years across hundreds of species. Natural selection has refined them, optimized them, turned them into reliable mechanisms for moving parasites through food webs. But there's something deeply unsettling about this. It challenges our assumptions about autonomy, about self-preservation, about the sanctity of individual life. We like to think that organisms, at least at some fundamental level, belong to themselves, that their actions serve their own interests. But parasites prove this isn't always true. An organism's actions can serve another's interests entirely. The host becomes an instrument, a vehicle, a temporary vessel whose existence is meaningful only insofar as it advances the parasite's goals. And yet, we shouldn't impose human values on these relationships. The parasite isn't evil. The host isn't a victim in any moral sense. These are just organisms following genetic programming, shaped by selection pressures we're only beginning to understand. The parasite does what increases its reproductive success. The host does what its altered nervous system tells it to do. There's no malice, no cruelty, no intention beyond the blind drive to propagate genes. Still, these examples force us to confront uncomfortable questions. If a parasite can make a cricket seek water against all instinct, if it can make a snail advertise itself to predators, if it can make an ant lock its jaws and wait for death, what does that say about the nature of will, about control, about the boundaries of self? We are, after all, biological organisms too. Our behaviors are produced by brains, by chemistry, by neurons firing in patterns. And if those patterns can be altered by external influences, whether parasites or drugs or environmental factors, then where exactly does our autonomous self reside? These aren't just philosophical questions, they're biological realities. Your brain right now is making you feel tired, relaxed, ready for sleep. Is that you choosing to rest? Or is it adenosine accumulating in your neurons, overriding your desire to stay awake? When you feel hungry and seek food, is that free will? Or is it ghrelin, a hormone produced by your stomach, compelling you toward the kitchen? The line between choice and compulsion is thinner than we imagine. Parasites that induce suicide in their hosts aren't doing anything fundamentally different from what your own body does to you every day. They're just using external chemistry instead of internal chemistry. They're hacking the same systems that normally govern behavior, the same neurotransmitters and hormones and neural circuits. The only difference is intent. Your body's signals generally serve your survival and reproduction. The parasite's signals serve its own. But the mechanism, the biological substrate, is the same. So as you drift closer to sleep tonight, consider this. Every impulse you feel, every desire, every decision, is mediated by chemistry. You are a chemical system, beautifully complex, but ultimately governed by molecules interacting according to physical laws. And systems can be influenced. Parasites prove this every day in countless species around the world. They prove that control is transferable, that behavior is hackable, and that the self is far more fragile and negotiable than we like to believe. Next, we'll bring this closer to home, exploring the parasites that infect mammals, including humans, and the subtle ways they might be shaping our thoughts, our personalities, and our choices right now. So far, we've been talking about parasites in insects, in fish, in snails. It's easy to think of these examples as distant, as belonging to a world separate from our own. But now, let's bring this closer. Let's talk about mammals. Let's talk about us. Because some of these mind-controlling parasites don't just infect crickets and ants. They infect cats, rats, pigs, sheep, and humans. Right now, as you're lying there listening, there's a reasonable chance you're carrying one of these parasites in your own brain, and it might be changing you in ways you've never noticed. Let's start with Toxoplasma gondii again, because this parasite doesn't just manipulate rodents. It infects an estimated 1/3 of the human population worldwide. That's over 2 billion people. In some countries, the infection rate is even higher, reaching 60 or 70%. Most people never know they're infected. The immune system keeps the parasite in check, contained within tissue cysts in the brain and muscles. There are usually no obvious symptoms. No fever, no pain, no visible signs. Just dormant cysts sitting quietly in your neurons, possibly for the rest of your life. But here's the question researchers have been asking for decades. If toxoplasma can alter behavior in rodents so dramatically, making them lose fear of predators, could it be doing something similar in humans? Could it be subtly changing our personalities, our risk tolerance, our mental health? The evidence is complicated, sometimes contradictory, but increasingly the answer seems to be yes, maybe. Multiple studies have found correlations between toxoplasma infection and personality traits. Infected individuals, on average, show slightly higher levels of risk-taking behavior. They're more likely to start businesses, more likely to engage in adventurous activities, more likely to have been in traffic accidents. Some research suggests infected men become more introverted, suspicious, and rule-breaking, while infected women become more outgoing, warm, and conscientious. The changes aren't dramatic. You wouldn't look at an infected person and immediately notice something different. But statistically, across large populations, patterns emerge. There's also research linking toxoplasma to mental health conditions. Several studies have found higher rates of infection in people with schizophrenia, bipolar disorder, and suicidal behavior. Again, correlation doesn't prove causation. It's possible that people with these conditions are more likely to become infected for other reasons, or that shared risk factors explain the association. But some researchers believe the parasite might actually contribute to mental illness by altering brain chemistry, particularly dopamine levels, which we know toxoplasma affects in rodents. One study found that people infected with toxoplasma had slower reaction times. When tested on simple cognitive tasks, infected individuals took slightly longer to respond. This could explain the association with traffic accidents. If your brain is processing information just a fraction of a second slower, that might be enough to miss a red light, to brake too late, to make a critical error in judgment. It's subtle, but in high-speed situations, subtle delays can be fatal. Now, before you start worrying, remember that most infected people live completely normal lives. The effects, if they exist at all, are small. You're not being turned into a zombie. You're not losing control of your actions. But the possibility that a microscopic parasite could be nudging your personality, shifting your risk tolerance by a few percentage points, altering your reaction time by a few milliseconds, that's genuinely fascinating and more than a little unsettling. And toxoplasma isn't the only parasite that might affect human behavior. Let's talk about rabies, one of the deadliest viruses on Earth. Rabies has a fatality rate of nearly 100% once symptoms appear, and the symptoms are distinctive. Infected animals, and occasionally humans, become aggressive, confused, and hypersalivate. They also develop hydrophobia, a fear of water so intense that even the sight or sound of water can trigger panic and throat spasms. Why would a virus make its host afraid of water? Because rabies spreads through saliva, usually via bites. The virus needs the host to bite other animals to transmit itself, but swallowing reduces the amount of virus-laden saliva in the mouth. So, the virus manipulates the nervous system to prevent swallowing, causing saliva to accumulate. At the same time, it increases aggression and confusion, making the host more likely to attack. It's behavioral manipulation designed to maximize transmission. The hydrophobia, the aggression, the biting, all of it serves the virus's reproductive strategy. In humans, rabies infection is rare, thanks to vaccines and animal control. But when it does occur, the behavioral changes are dramatic and tragic. People become paranoid, hallucinate, experience uncontrollable aggression and fear. Their personality disintegrates as the virus destroys brain tissue. It's one of the clearest examples of a pathogen completely overriding human behavior, turning a person into something unrecognizable. And it's a reminder that our brains, for all their complexity, are vulnerable to biological hijacking. There are other examples, less dramatic but potentially more common. Some intestinal parasites have been linked to fatigue, depression, and cognitive impairment. The mechanisms aren't fully understood, but chronic inflammation caused by parasitic infections can affect brain function. Inflammatory cytokines can cross the blood-brain barrier and alter neurotransmitter metabolism. The result might be brain fog, mood changes, difficulty concentrating. Not direct manipulation like we see with toxoplasma, but indirect effects that still change how you think and feel. Even gut bacteria, which aren't parasites but are still foreign organisms living inside you, have been shown to influence mood and behavior. The gut-brain axis, the communication network between your digestive system and your central nervous system, allows microbes in your intestines to send signals that affect anxiety, depression, and even social behavior. Some bacteria produce neurotransmitters like serotonin and GABA. Others release metabolites that influence brain chemistry. You are, in a very real sense, a collaborative organism. Your thoughts and feelings aren't produced by your neurons alone, but by a community of organisms, some human, some microbial, all interacting in ways we're only beginning to map. This raises profound questions about identity and agency. If a parasite in your brain is producing dopamine, and that dopamine is influencing your decisions, whose decisions are they? If bacteria in your gut are affecting your mood, is that mood authentically yours? Where do you end and the microbial world begins? These aren't just philosophical puzzles. They're practical questions with implications for how we understand mental health, personality, and consciousness itself. Some researchers are now exploring whether treating parasitic infections could alleviate certain psychiatric symptoms. If toxoplasma is contributing to schizophrenia or bipolar disorder, could antiparasitic drugs help? Some preliminary studies suggest they might, though the evidence is still limited and controversial. It's too early to say definitively. But the possibility that mental illness could be partly infectious, caused or worsened by parasites, is a paradigm shift in psychiatry. And then there's the flip side. Some parasitic infections might actually be protective. There's a hypothesis called the hygiene hypothesis, which suggests that reduced exposure to parasites and other microbes in modern, sanitized environments has led to increases in allergies, autoimmune diseases, and possibly even some mental health conditions. Our immune systems evolved in environments rich with parasites. Some researchers believe we might need a certain level of parasitic exposure to calibrate our immune responses properly. Remove all parasites, and the immune system might become overactive, attacking the body's own tissues, or overreacting to harmless substances like pollen. Some studies have even tested therapeutic parasitic infections, intentionally infecting people with hookworms or whipworms to treat conditions like Crohn's disease and ulcerative colitis. The results have been mixed, but in some cases, people experience significant improvement. The parasites seem to modulate the immune system, reducing inflammation and symptoms. It's a controversial approach, but it highlights how complex our relationship with parasites really is. They're not just enemies, they're part of the ecological context in which we evolved. So what does all this mean for you, lying there tonight, possibly hosting a few parasites of your own? First, don't panic. Most parasitic infections in developed countries are mild or asymptomatic. Your immune system is doing its job, keeping populations in check. Second, recognize that you're not a closed system. You're an ecosystem. You contain multitudes, bacteria, viruses, fungi, maybe a few parasites, and those organisms are part of what makes you, you. They influence your digestion, your immune function, possibly your mood and behavior. Third, appreciate the complexity. The fact that a microscopic organism can influence human personality is both humbling and awe-inspiring. It shows how deeply interconnected life is, how biology operates across scales from molecules to ecosystems, and how much we still have to learn about the systems that govern our own minds. You are not just a brain in a body. You are a community, a negotiation between different genomes, different evolutionary interests, all coexisting in the vessel you call yourself. And finally, rest easy, because whether or not you have toxoplasma cysts in your brain, Whether or not your gut bacteria are tweaking your serotonin levels, you're still here, still thinking, still capable of choice and reflection. Biology shapes us, yes, but it doesn't define us completely. We are the product of chemistry and evolution, but also of experience, learning, and culture. We contain multitudes, literally and figuratively. Next, we'll zoom out and look at the bigger picture, the evolutionary arms race between parasites and hosts, and how this endless conflict has shaped the diversity of life on Earth. Imagine two armies locked in an eternal battle. Neither side can ever fully win. Every advance by one is eventually countered by the other. Every weapon developed triggers the evolution of new armor. Every defense created leads to the invention of new attack. This war has no end, no peace treaty, no final victory. It just continues, generation after generation, driving both sides to become more sophisticated, more specialized, more extreme. This is the evolutionary arms race between parasites and hosts, and it's been shaping life on Earth for hundreds of millions of years. Let's start with defense. Hosts don't just passively accept parasitic infection, they fight back. And the first line of defense is the immune system, an incredibly complex network of cells, proteins, and chemical signals designed to recognize and destroy foreign invaders. In mammals, this system includes physical barriers like skin and mucous membranes, cellular defenses like white blood cells that engulf and digest pathogens, and adaptive immunity that remembers specific threats and responds faster on subsequent exposures. Your immune system is constantly surveilling your body, checking every cell to make sure it's supposed to be there. Specialized cells called T cells and B cells patrol your tissues, looking for molecular signatures that indicate infection. When they find something foreign, a parasite protein that doesn't belong, they sound the alarm. Inflammation begins. More immune cells are recruited to the site, antibodies are produced, Y-shaped proteins that bind to parasites and mark them for destruction. The infected area might swell, turn red, heat up. That's your body mobilizing resources to eliminate the threat. But immune systems aren't perfect. They can be fooled, overwhelmed, or misdirected. And parasites have evolved countless ways to evade them. We've already talked about molecular mimicry, where parasites coat themselves in host proteins to avoid recognition, but there are other strategies too. Some parasites hide inside cells, where they're invisible to circulating antibodies. Others rapidly change their surface proteins, presenting a moving target that the immune system can't lock onto. It's like trying to hit a shapeshifter that never looks the same twice. Trypanosomes, the parasites that cause sleeping sickness in humans and livestock, are masters of this technique. They have genes for hundreds of different surface proteins, and they can switch between them. Your immune system recognizes one variant, produces antibodies against it, and starts to clear the infection. But before the parasites are eliminated, some of them switch to a different surface protein. The antibodies no longer work. The immune system has to start over, recognizing the new variant and producing new antibodies. By the time it does, the parasites have switched again. This process can continue for months or years, waves of parasites appearing and being controlled, then reappearing in a different disguise. Hosts have evolved behavioral defenses too. Grooming is one of the most important. Primates spend hours each day picking through each other's fur, removing parasites, dead skin, and debris. Birds preen their feathers. Cats lick themselves obsessively. This isn't just vanity, it's parasite control. By physically removing parasites before they can establish infections, grooming reduces parasite load and transmission. Some species even use tools. Chimpanzees have been observed using rough leaves to scrub parasites from their skin. Birds take dust baths or ant baths, using the formic acid from ants to kill mites and lice. Social immunity is another fascinating defense. In insect colonies, individuals groom each other, removing fungal spores and parasitic mites. When one individual becomes infected, others may isolate it, preventing the infection from spreading through the colony. Termites and ants produce antimicrobial compounds that they spread throughout their nests, creating a chemical barrier against fungal and bacterial parasites. Bees perform hygienic behavior, detecting and removing infected larvae before they can contaminate the hive. The colony functions as a single organism, with distributed immune responses coordinated through chemical communication. But for every defense, parasites evolve counter-defenses. If hosts develop grooming behavior, parasites evolve to hide in places that are hard to reach. If hosts isolate infected individuals, parasites evolve to suppress symptoms during the infectious stage, allowing hosts to move freely and spread the infection before becoming visibly sick. If hosts produce antimicrobial compounds, parasites evolve resistance. The arms race escalates. And here's where things get really interesting. Parasites often evolve faster than their hosts. Why? Because they have shorter generation times. A mouse might reproduce once or twice a year. Toxoplasma can go through multiple generations in a matter of weeks. This means parasites can adapt to host defenses much more quickly than hosts can evolve new defenses. It's an asymmetry that gives parasites a significant advantage. They're operating on evolutionary fast forward. But hosts have an advantage too: sexual reproduction. When organisms reproduce sexually, they shuffle their genes, creating offspring with new combinations of traits. This genetic diversity makes it harder for parasites to adapt. A parasite that's well suited to infect one individual might be less effective against that individual's offspring, who inherited a different mix of immune genes. Sexual reproduction is essentially a bet hedging strategy. You don't know which parasite variants your offspring will encounter, so you give them a diverse genetic portfolio, hoping something will be effective. This idea is called the Red Queen hypothesis, named after the character in Lewis Carroll's Through the Looking Glass, who tells Alice, "It takes all the running you can do to keep in the same place." In evolutionary terms, hosts must constantly evolve just to maintain their current level of resistance, because parasites are constantly evolving new ways to infect them. Neither side gets ahead permanently. They're locked in a dynamic equilibrium, where change is necessary simply to avoid extinction. There's evidence for this hypothesis across many species. Populations with high parasite pressure tend to have more sexual reproduction, more genetic diversity in immune genes, and faster evolutionary rates. In laboratory experiments, when parasites are allowed to co-evolve with hosts, both sides show rapid adaptation. The hosts become more resistant. The parasites become more virulent or better at evasion. Remove the parasites, and host evolution slows dramatically. The arms race drives change on both sides. Some of the most important genes in the human genome are involved in immune function. The major histocompatibility complex, or MHC, is a group of genes that code for proteins involved in recognizing foreign molecules. These genes are among the most diverse in the entire human genome. Why? Because parasites create selection pressure for diversity. Individuals with rare MHC variants are less likely to succumb to common parasites, which haven't yet adapted to those variants. Over time, this maintains a huge variety of MHC genes in the population, its genetic diversity driven by parasitic pressure. And this diversity has consequences beyond immunity. Some research suggests that MHC genes influence mate choice. People are attracted to partners with different MHC profiles, possibly because offspring from such pairings will have more diverse immune genes and better resistance to parasites. You might be choosing your romantic partners, in part, based on unconscious assessments of immune compatibility. It sounds strange, but the evidence is accumulating. Parasites might be shaping not just our biology, but our social behavior and reproductive decisions. The arms race also drives the evolution of complexity. As parasites develop more sophisticated ways to evade immunity, hosts evolve more sophisticated immune systems. As hosts develop better defenses, parasites evolve more elaborate life cycles involving multiple hosts and complex manipulation strategies. Complexity begets complexity. The result is the astonishing diversity of parasitic strategies we've been exploring, and the equally astonishing diversity of host defenses. But here's the paradox. Despite all this conflict, neither side wants to completely eliminate the other. If a parasite becomes too virulent, killing hosts too quickly, it reduces its own transmission opportunities. Dead hosts don't spread parasites. So there's selection pressure for moderation, for parasites that keep hosts alive long enough to reproduce and transmit. Similarly, if a host becomes completely immune to all parasites, it might seem like a good thing. But remember the hygiene hypothesis. Immune systems calibrated in the absence of parasites might malfunction, attacking the body's own tissues or overreacting to harmless stimuli. A certain level of parasitic infection might actually be beneficial or at least necessary for proper immune development. This leads to an idea called the tolerance model. Instead of focusing only on resistance, eliminating parasites, some hosts might benefit from tolerating low levels of infection. Tolerance means reducing the damage caused by infection without necessarily reducing the parasite load. It's a different strategy, coexistence rather than eradication, and in some cases, it might be more effective. Fighting off parasites is energetically expensive. It requires constant immune surveillance, inflammation, tissue repair. If you can tolerate the parasites without suffering too much damage, you save energy that can be invested in growth, reproduction, or other survival needs. The arms race, then, isn't always about winning. It's about balance, finding an equilibrium where both host and parasite can coexist. Not peacefully, not harmoniously, but sustainably. The host survives long enough to reproduce. The parasite transmits successfully enough to persist. And the ecosystem as a whole maintains its structure, with parasites playing their role in regulating populations, transferring energy, and driving evolutionary change. So when you think about infection, about disease, about the parasites that might be living inside you right now, remember that you're part of an ancient story, a story that began billions of years ago, when the first organisms started exploiting each other. A story that's written in your genome, in your immune system, in the very structure of your cells. You are the product of countless generations of conflict and coexistence, shaped by parasites as much as by any other force in nature. Next, we'll explore just how many parasites are out there, the hidden biodiversity that most people never think about, and the essential roles parasites play in keeping ecosystems stable and functional. Close your eyes for a moment and picture a forest. What do you see? Trees, probably. Maybe some deer grazing, birds in the canopy, a stream running through, mushrooms on a log, insects buzzing. That's the visible ecosystem, the part we notice, the part that feels alive and dynamic. But there's another ecosystem hidden within it, an invisible kingdom that most people never consider. Parasites. Thousands of species, in every animal, in every plant, in every body of water. If you could see them all at once, the landscape would transform. Every organism glowing with the countless smaller organisms living inside it. The forest wouldn't look empty. It would look impossibly full. Here's a statistic that might surprise you. Scientists estimate that parasites may represent up to half of all species on Earth. Half. That means for every free-living organism you can name, a tree, a bird, a fish, there's at least one parasite species that specializes in infecting it. And often, there are many more than one. A single host species might harbor dozens or even hundreds of different parasitic species, each one occupying a different niche, targeting different tissues, using different transmission strategies. Let's take a single animal as an example, a deer. If you examine that deer thoroughly, inside and out, you'd find tapeworms in its intestines, flukes in its liver, nematodes in its muscles, botfly larvae under its skin, ticks attached to its hide, mites in its ears, and protozoans in its blood. Each one of these parasites is a species with its own life cycle, its own evolutionary history, its own ecological relationships. And that's just one deer. Multiply that by every animal in the forest, every fish in the stream, every bird in the sky, and you begin to appreciate the sheer scale of parasitic biodiversity. But parasites aren't just numerous. They're also incredibly diverse in form and function. We've talked about fungi, worms, protozoans, viruses, bacteria, but even within these groups, the variation is staggering. Tapeworms can grow to 10 meters long inside a human intestine, while some parasitic protozoans are smaller than a red blood cell. Some parasites live for decades inside a single host. Others complete their entire life cycle in a matter of days. Some manipulate behavior. Others simply steal nutrients. Some kill their hosts quickly. Others keep them alive for years. And they're everywhere. Every ecosystem on Earth, from the deepest ocean trenches to the highest mountain peaks, from tropical rainforests to Arctic tundra, contains parasites. There are parasites that infect bacteria, parasites that infect parasites, layers upon layers of exploitation, cascading through the web of life. You cannot find a habitat, no matter how extreme, that doesn't host parasitic organisms. They've colonized every possible niche. Now, you might be thinking, if parasites are so common, they must be doing something important. And you'd be right. For a long time, ecologists viewed parasites as incidental, side characters in the story of ecosystems. The real players were predators and prey, plants and herbivores, the visible interactions that shaped communities. But over the last few decades, that view has changed. Scientists now recognize that parasites are fundamental to ecosystem function. They're not just passengers, they're drivers. Let's start with population control. Parasites regulate host populations by causing disease and death. This prevents any single species from becoming too dominant. In a forest where deer have no predators, their population can explode, overgrazing vegetation and degrading habitat. But parasites act as a check. They spread more easily in dense populations, where hosts are in close contact. As deer numbers increase, so does parasite transmission. Disease outbreaks thin the herd, bringing populations back down. It's a natural form of population regulation, operating invisibly but powerfully. Parasites also influence competition between species. If two species compete for the same resources, the one with fewer parasites often has an advantage. It's healthier, stronger, better able to compete. This can shift the balance, allowing one species to dominate while the other declines. In some cases, parasites have been shown to maintain biodiversity by preventing competitive exclusion. They infect the dominant species more heavily, weakening it enough that subdominant species can coexist. Without parasites, ecosystems would be simpler, less diverse, more prone to dominance by a few highly successful species. Energy flow is another critical role. We've already seen how hairworms transfer energy from terrestrial ecosystems to aquatic ones by driving infected insects into water. This kind of cross-ecosystem energy transfer happens constantly. Parasites move nutrients and energy between habitats, between trophic levels, in ways that wouldn't occur otherwise. When a bird eats an infected snail, it's not just consuming the snail's tissue. It's also consuming the parasite's biomass, which represents energy that was originally harvested by the snail but redirected into parasite growth. Parasites are essentially recyclers, concentrating and redistributing resources through ecosystems. And then there's the role parasites play in food web stability. Food webs are networks of who eats whom, but traditional food webs usually ignore parasites. When you include them, the structure becomes far more complex. Parasites connect species that wouldn't otherwise interact. They create hidden links between predators and prey, between herbivores and plants. And these links stabilize the web. Research has shown that ecosystems with more parasite diversity are more resilient to disturbances. They recover faster from perturbations. They're less likely to collapse when key species are removed. Why? Because parasites create redundancy. If one pathway for energy flow is disrupted, parasites provide alternative routes. They also dampen oscillations. Predator-prey systems can be unstable, with populations cycling boom and bust. Parasites smooth out these cycles by independently regulating both predator and prey populations. They're like shock absorbers, preventing the extreme swings that can lead to extinction. Now imagine what would happen if you removed all parasites from an ecosystem. It's a thought experiment, but a revealing one. Initially, it might seem like a good thing. Animals would be healthier, plants would be free from fungal infections, populations would grow. But, very quickly, problems would emerge. Without parasitic regulation, some species would explode in number, overconsuming resources. Competition would intensify. Dominant species would exclude others, biodiversity would decline, energy flows would become less efficient. The web would simplify, and simplified ecosystems are fragile ecosystems. They're vulnerable to disease, to climate change, to invasive species. Parasites, it turns out, are essential threads in the fabric of life. Pull them out, and the whole thing unravels. There's also a genetic contribution. Parasites maintain genetic diversity in host populations. We talked about this in the context of the Red Queen hypothesis, but it's worth emphasizing. Because parasites create selection pressure for diversity, they prevent populations from becoming genetically uniform. And genetic diversity is insurance. It means that when environments change, when new threats emerge, there's variation in the population for natural selection to act on. Some individuals will have the right combination of genes to survive. Without parasites driving diversity, populations would be less adaptable, more prone to extinction when conditions shift. But here's the challenge. Parasites are disappearing. Not all of them, but many. As humans alter landscapes, fragment habitats, and drive host species to extinction, we're also causing parasite extinctions. And these losses are largely invisible. No one holds fundraisers to save parasitic worms. No conservation campaigns focus on preserving tapeworm diversity. But ecologically, these extinctions might be just as damaging as losing a charismatic megafauna species like tigers or elephants. Some scientists argue that we need parasite conservation, that we need to recognize the value of these organisms and protect them just as we protect other components of biodiversity. It's a hard sell, emotionally and politically, but scientifically it makes sense. Parasites are part of the natural heritage of every ecosystem. They've co-evolved with their hosts for millions of years. They perform functions we're only beginning to understand. Losing them means losing ecological complexity, stability, and resilience. And we're losing them faster than we can document them. Most parasite species have never been formally described by science. They exist in the tissues of host species we've barely studied, in ecosystems we're rapidly destroying. By the time we realize they're important, many will be gone. It's a silent extinction crisis, happening beneath our notice in the hidden kingdom we've spent tonight exploring. So, when you think about biodiversity, expand your definition. Don't just count the trees and the birds and the mammals. Count the parasites too. They're the majority. They're the hidden workforce, regulating, connecting, stabilizing. They're the engineers of complexity, the architects of ecological relationships, and they're part of what makes the natural world function. Not in spite of them, but because of them. The ecosystem isn't just the hosts, it's the parasites too, and we're all richer for their presence, whether we realize it or not. Next, we'll turn back the clock and explore the deep history of parasitism, tracing these relationships back through evolutionary time to understand how this way of life first began and how it shaped the entire tree of life. Let's go back, way back, not just centuries or millennia, but hundreds of millions of years, to a time when the Earth looked nothing like it does today. The continents were arranged differently, the climate was warmer, and life was simpler, but already experimenting with one of nature's most enduring strategies: parasitism. The question scientists have long wrestled with is this: when did parasitism begin? How did free-living organisms first make the transition to living inside or on other organisms? And what can the ancient origins of parasitism tell us about evolution itself? The fossil record of parasites is frustratingly incomplete. Soft-bodied organisms don't fossilize well. A tapeworm, a fluke, a protozoan, these leave almost no trace.
So most of what we know about ancient parasitism comes from indirect evidence.
Bite marks on fossil bones, deformities in ancient shells that suggest infection, and occasionally, extraordinarily preserved specimens that capture parasites in the act. One of the most famous examples comes from the Messel Pit in Germany, a fossil site that preserves organisms from about 47 million years ago. Scientists have found fossilized leaves with galls, those parasite-induced plant growths we talked about earlier. The galls are perfectly preserved, showing that even 47 million years ago, insects were manipulating plants to build nurseries for their larvae. But parasitism is much, much older than that. Molecular clock studies, which use genetic differences between species to estimate when they diverged, suggest that many parasitic lineages are hundreds of millions of years old. Some parasitic groups may have originated over 500 million years ago during the Cambrian explosion, that period of rapid evolutionary diversification when most major animal groups first appeared. If parasites were present then, it means they've been part of ecosystems for nearly as long as complex multicellular life itself. So how did it start? Imagine an ancestral organism, free living, perhaps feeding on bacteria or decaying organic matter. One day, through random mutation or behavioral variation, one individual starts spending time near another, larger organism. Maybe it feeds on shed skin cells. Maybe it consumes mucus or other secretions. This isn't parasitism yet. It's just opportunistic feeding. But over generations, if this strategy proves successful, natural selection might favor individuals that stay closer to the host, that develop specialized mouthparts for feeding on host tissue, that evolve ways to avoid being dislodged or eaten. Gradually, the relationship deepens. The organism becomes more dependent on the host. Eventually, it can no longer survive independently. It's crossed the line into obligate parasitism. This transition has happened independently countless times across the tree of life. Parasitism isn't a single invention. It's a strategy that different lineages have discovered over and over. Flatworms gave rise to parasitic flukes and tapeworms. Roundworms gave rise to countless parasitic nematodes. Crustaceans gave rise to parasitic barnacles like Sacculina. Even vertebrates have parasitic representatives. Lampreys, for example, are parasitic fish that attach to other fish and feed on their blood and tissues. Parasitism, it seems, is an evolutionary attractor. Given enough time and opportunity, many lineages will stumble into it. One of the key transitions in parasitic evolution is the loss of traits. When you become a parasite, you often don't need the abilities your ancestors had. If you're living inside a host, feeding on pre-digested nutrients, you don't need a complex digestive system. You can lose your gut. If you're not moving around much, you don't need well-developed sense organs or a sophisticated nervous system. You can simplify. This is called regressive evolution, and it's common in parasites. Tapeworms, for example, have no digestive system at all. They absorb nutrients directly through their skin. Sacculina has lost nearly every recognizable feature of a barnacle. It's been reduced to a reproductive sac and a root system. When your environment is another organism's body, you can afford to specialize, to shed the baggage of traits you no longer need. But parasites also evolve new traits, structures for attaching to hosts, hooks, suckers, adhesive organs, mechanisms for evading immune systems, complex life cycles involving multiple hosts, behavioral manipulation abilities. These are innovations, adaptations that only make sense in the context of a parasitic lifestyle. So parasites are simultaneously regressing, losing old traits, and progressing, gaining new ones. It's a process of evolutionary refinement, streamlining for a highly specialized niche. Phylogenetic studies, which reconstruct evolutionary relationships by comparing DNA sequences, have revealed some surprising origins. Many parasitic groups are nested within free-living groups, showing that the transition to parasitism happened relatively recently in evolutionary terms. For example, parasitic wasps are closely related to free-living wasps. The shift to laying eggs inside other insects rather than in nests happened within the wasp family tree. Similarly, parasitic plants like mistletoe are related to free-living plants. They evolved from ancestors that had normal roots and photosynthesis, but gradually became dependent on tapping into the vascular systems of host trees. And once a lineage becomes parasitic, it often diversifies rapidly. This is because parasitism opens up new ecological opportunities. Each host species represents a potential niche. If you can adapt to infect a new host, you've essentially colonized a new habitat. Over time, parasitic lineages radiate, splitting into multiple species, each specialized for different hosts or different tissues within hosts. This is why there are so many parasite species. Every host is an island, and evolution fills islands with diversity. Some of the most ancient parasitic relationships we know about involve single-celled organisms. Apicomplexa, the group that includes Plasmodium, the malaria parasite, and Toxoplasma, are thought to have originated over 1 billion years ago. These parasites have incredibly complex cell structures, with specialized organelles for invading host cells. Their ancestors were likely free-living algae, photosynthetic organisms that lived in water. At some point, they lost their chloroplasts, the structures that perform photosynthesis, and transitioned to feeding on other cells. Over hundreds of millions of years, they became the highly specialized parasites we see today. Tracing these origins is like detective work. Scientists compare genomes, looking for remnants of lost traits. In Apicomplexa, for example, researchers have found vestigial genes related to photosynthesis. These genes no longer function, but they're still there. Molecular fossils that hint at the parasite's photosynthetic past. It's as if evolution leaves a trail, breadcrumbs we can follow back to ancestral states. Co-evolution is another critical aspect of parasitic history. Parasites and hosts don't evolve in isolation. They shape each other. When a host evolves a new defense, parasites that can overcome that defense are favored. When a parasite evolves a new virulence factor, hosts that can resist it survive better. This reciprocal evolution creates patterns in the fossil record and in genetic data. Parasite phylogenies often mirror host phylogenies. When a host species splits into two, its parasites often split too, each lineage following its respective host. This is called cophylogeny, and it's evidence of deep, ancient partnerships. For example, lice that infect primates have a phylogeny that closely matches the primate phylogeny. Human lice are most closely related to chimpanzee lice, gorilla lice, and so on, reflecting the evolutionary relationships among the hosts. This tells us that lice have been parasitizing primates for millions of years, co-evolving with us as we diverge from our common ancestors. Your head lice, annoying as they are, carry a history that predates humanity itself. And this raises a humbling thought. Every organism alive today, including you, is the product of countless encounters with parasites. Your immune system, your behavior, even your genome have been shaped by parasitic pressure. The ancestors that survived to pass on their genes were the ones who could resist, tolerate, or avoid parasites. The ones who couldn't died out. Parasites have been selecting traits in their hosts for as long as the relationship has existed. And hosts have been selecting traits in parasites just as long. It's a mutual sculpting, each side shaping the other's evolution. Some researchers argue that parasitism might have played a role in major evolutionary transitions. The evolution of sexual reproduction, for example, might have been driven partly by the need to generate genetic diversity to combat parasites. The evolution of multicellularity might have been influenced by the pressure to create barriers against single-celled parasites. The evolution of immune systems was certainly shaped by parasitic infections. In a very real sense, parasites have been architects of complexity, pushing their hosts to evolve new structures, new behaviors, new strategies for survival. And parasites themselves have contributed genetic material to their hosts. Horizontal gene transfer, where genes move between species, is common in microbes but also occurs between parasites and hosts. Some of the genes in your genome may have originally come from viruses or bacteria that infected your distant ancestors. These foreign genes were incorporated, co-opted, and now serve new functions. Your body contains genetic echoes of ancient infections, molecular memories of parasites long gone but not forgotten. So when you think about your own evolutionary history, remember that you're not just descended from a line of successful predators, or clever tool users, or social cooperators. You're also descended from survivors of countless parasitic infections. Every generation of your ancestors faced parasites. And the fact that you're here, alive and listening, means they won those battles often enough to reproduce. You are the product of an ancient war, a war that's still being fought in your body and in everybody, every moment of every day. Next, we'll look forward instead of back, exploring how modern science is learning from parasites and applying their strategies to medicine, technology, and our understanding of the mind itself. Parasites have spent hundreds of millions of years perfecting the art of infiltration, evasion, and control. They've learned how to cross biological barriers, manipulate immune systems, and deliver molecular payloads with surgical precision. And now, human scientists are starting to ask a fascinating question. Can we learn from them? Can we take the strategies parasites use and repurpose them for medicine, for technology, for treating diseases that have resisted every other approach? The answer, increasingly, is yes. Parasites are becoming teachers, and the lessons they're offering could transform how we think about drug delivery, immunology, and even neurological disorders. Let's start with drug delivery. One of the biggest challenges in medicine is getting drugs to the right place in the body. You can develop a perfect molecule, something that kills cancer cells or repairs damaged neurons, but if you can't deliver it to the target tissue, it's useless. Drugs taken orally get broken down in the stomach. Drugs injected into the bloodstream get filtered out by the liver and kidneys. And the blood-brain barrier, that protective shield around your central nervous system, blocks most large molecules from entering the brain. This is why so many neurological diseases are hard to treat. The drugs simply can't reach the neurons that need them. But parasites cross the blood-brain barrier all the time. Toxoplasma does it. So do certain species of trypanosomes and nematodes. They've evolved molecular mechanisms for breaching this barrier without causing massive inflammation or damage. So researchers are studying these mechanisms, trying to understand how parasites do it. What proteins do they use? What signals do they send? Can we mimic those signals and use them to smuggle therapeutic drugs across the barrier? Some early experiments suggest this might work. Scientists have designed nanoparticles coated with parasite-derived proteins. These particles can cross the blood-brain barrier, carrying drugs with them, and deliver treatments directly to brain tissue. It's still experimental, but the potential is enormous. Brain tumors, Alzheimer's disease, Parkinson's, conditions that have been nearly untreatable might become manageable if we can solve the delivery problem. Parasites are also teaching us about immune evasion. We talked earlier about how parasites coat themselves in host molecules to avoid detection. This is essentially stealth technology, biological camouflage, and it has applications beyond parasitology. Researchers working on organ transplants face a similar problem. When you transplant a kidney or a heart,
The recipient's immune system recognizes it as foreign and attacks it. Patients have to take immunosuppressive drugs for the rest of their lives, which leave them vulnerable to infections and other complications. But what if you could coat the transplanted organ in molecules that make it look like self? Parasite-inspired camouflage could reduce or eliminate the need for immunosuppression, making transplants safer and more successful. Similar principles apply to artificial organs and medical implants. Your body often rejects synthetic materials, forming scar tissue around them or triggering chronic inflammation. But if those materials could be coated with parasite-like molecular disguises, they might integrate more smoothly, accepted by the body as if they belong there all along. It's biomimicry at the molecular level, learning from organisms that have solved problems we're still struggling with.
Then there's the question of immunomodulation. Parasites don't just evade the immune system.
They actively manipulate it, turning down inflammation, redirecting immune responses, creating environments where they can thrive. And some of these manipulations might actually be beneficial for humans with certain conditions. We touched on this earlier with the hygiene hypothesis and therapeutic parasitic infections. But let's go deeper. There are now clinical trials using molecules derived from parasitic worms to treat autoimmune diseases like multiple sclerosis, rheumatoid arthritis, and inflammatory bowel disease. The idea is simple. These diseases are caused by overactive immune systems attacking the body's own tissues. Parasites produce molecules that calm immune responses. So if you can isolate those molecules, synthesize them in a lab, and give them to patients, you might be able to reduce inflammation without the risks of traditional immunosuppressive drugs. Some of these parasite-derived compounds are showing promise. They're specific, targeting only certain immune pathways. They're potent, working at low doses, and they're relatively safe, at least in early trials. It's too soon to say whether they'll become standard treatments, but the principle is sound. Parasites have been managing immune systems for millions of years. We're just catching up. Behavioral manipulation is another frontier. Understanding how parasites alter behavior could help us treat neurological and psychiatric disorders. If we can figure out exactly which neural circuits toxoplasma targets to reduce fear, we might be able to design therapies for anxiety disorders or PTSD. If we can understand how zombie ant fungi control motor neurons, we might develop better treatments for movement disorders like Parkinson's. The brain is still largely a black box. We know what regions do generally, but the fine details of how thoughts, emotions, and behaviors are generated remain mysterious. Parasites, through their manipulations, are giving us clues. They're inadvertent neuroscientists, running experiments that humans could never ethically perform. There's also the possibility of using parasitic strategies for pest control. Right now, agriculture relies heavily on chemical pesticides, which harm non-target species, pollute waterways, and lose effectiveness as pests evolve resistance. But parasites offer a biological alternative. Many insects have natural parasites that regulate their populations. By introducing these parasites to agricultural systems, we can control pest populations without chemicals. This is called biological control, and it's been used successfully in some contexts. For example, parasitic wasps have been released to control aphid populations in greenhouses. The wasps lay eggs inside the aphids, killing them before they can reproduce. It's targeted, sustainable, and doesn't create the environmental problems associated with chemical pesticides. But there are risks. Introducing parasites to new environments can have unintended consequences. The parasite might infect non-target species.
it might disrupt existing ecological relationships. There are famous examples of biological control gone wrong, where introduced species became invasive and caused more harm than the original pest. So this approach requires careful research, modeling, and monitoring. But when done correctly, it's a powerful tool, one that leverages nature's own pest control mechanisms. Then there's the philosophical and technological frontier. Understanding parasitic control raises questions about consciousness, free will, and the nature of self. If a microscopic organism can alter behavior by tweaking a few neurotransmitters, what does that say about human agency? And could we, in principle, use similar techniques to alter our own behavior? Not through infection, but through targeted brain stimulation, through drugs, through genetic modification. This edges into neuroethics, the study of the moral implications of neuroscience. If we develop the ability to manipulate our own brains the way parasites manipulate their hosts, how should we use that power? To treat disease, certainly. But what about enhancement? What about changing personality, reducing aggression, increasing empathy, altering preferences? These aren't hypothetical questions. We already have drugs that change mood, attention, and behavior. Antidepressants, stimulants, anti-anxiety medications. We're already intervening in brain chemistry. The difference is degree and precision. As our understanding deepens, as we learn more from parasites and other natural systems, our interventions will become more targeted, more effective, and more profound. And that will require careful thought about what we're trying to achieve, about the trade-offs between autonomy and well-being, about what it means to be human in a world where the boundaries of self are increasingly negotiable. Parasites are also inspiring advances in material science. The hooks and suckers parasites used to attach to hosts are incredibly strong and durable. They work in wet environments, they resist immune attacks, and they don't trigger excessive inflammation. Engineers are studying these attachment mechanisms to design better surgical adhesives, bandages that stick in the presence of blood, and coatings for medical devices. Some parasites produce silk-like proteins stronger than spider silk. Others secrete adhesives that work underwater. By understanding the chemistry and structure of these materials, we can create synthetic versions with applications far beyond medicine. Construction, manufacturing, even space exploration could benefit from parasite-inspired materials. And finally, parasites are helping us understand evolution itself. They're model systems for studying rapid adaptation, co-evolution, and the dynamics of host-parasite interaction. The principles we learn from parasites apply broadly: how organisms respond to environmental change, how complexity arises, how cooperation and conflict shape biological systems. Parasites are laboratories, natural experiments running in real time, generating data that informs not just medicine and ecology, but fundamental biology. So when you think about the future of science and medicine, remember that some of the most important innovations might come from the strangest sources. From worms that make crickets drown themselves, from fungi that turn ants into zombies, from single-celled organisms that cross barriers we can't. Nature has already solved many of the problems we're grappling with. We just need to pay attention, to study these solutions, and to adapt them for our own purposes. Parasites aren't just threats, they're teachers, and the lessons they're offering could change everything. Now, as we approach the end of our journey, let's step back and consider the bigger picture, the philosophy of parasitism, and what it means to live in a world where exploitation and cooperation are two sides of the same coin. We've spent the last few hours exploring parasites, their biology, their strategies, their ancient origins, and their potential futures. We've met organisms that hijack brains, transform bodies, and orchestrate deaths. We've seen how parasites shape ecosystems, drive evolution, and might even be influencing human personality. But now, as we reach the end of this journey together, let's step back and ask a different kind of question. Not what parasites do, but what they mean. What can they teach us about life itself, about cooperation and conflict, about the nature of relationships, and about our place in the vast web of existence? Let's start with a simple observation. We've been using the word parasite as if it has a clear definition, an organism that lives on or in another organism, causing harm while benefiting itself. But the more closely you look, the blurrier that definition becomes. Is a parasite that causes minimal harm really a parasite? Or is it a commensal, an organism that benefits without significantly helping or harming its host? And what about organisms that start as parasites but eventually become beneficial? Where exactly is the line? Consider your mitochondria, those tiny powerhouses inside every cell in your body. They generate the energy you need to live, to think, to move. But mitochondria have their own DNA, separate from your nuclear DNA. They reproduce independently, and that's because they weren't always part of you. Billions of years ago, they were free-living bacteria. At some point, they were engulfed by an ancestral cell, possibly as a parasitic infection. But instead of destroying the host or being destroyed, they formed a partnership. The bacteria provided energy. The host cell provided protection and resources. Over time, they became inseparable. What started as parasitism became symbiosis and eventually became you. You are, in a very literal sense, the descendant of a successful parasitic relationship. This pattern repeats throughout biology. Many of the most important evolutionary innovations began as parasitic or antagonistic relationships that gradually became cooperative. Chloroplasts, the structures in plant cells that perform photosynthesis, were once free-living cyanobacteria. Lichens are partnerships between fungi and algae, relationships that probably began as fungal parasitism but evolved into mutualism. Even the placenta, that organ that nourishes developing mammalian embryos, contains genes derived from ancient viruses that infected our ancestors. Parasitism and cooperation aren't opposites. They're points on a spectrum, and organisms can move along that spectrum over evolutionary time. So, when we label something a parasite, we're making a judgment about harm and benefit at a specific moment in time. But from a longer evolutionary perspective, today's parasite might be tomorrow's symbiont. The relationship isn't fixed. It's dynamic, constantly being renegotiated by natural selection. And this fluidity challenges our moral intuitions. We tend to see parasites as bad, as villains exploiting innocent hosts. But nature doesn't deal in morality, it deals in consequences. If a relationship persists, it's because both parties, or at least their lineages, are gaining something, even if that gain comes at a cost. Let's think about this from the host's perspective. Yes, parasites cause harm. They steal resources, damage tissues, manipulate behavior, but hosts aren't passive victims. They have defenses, they evolve resistance, and in many cases they tolerate parasites because the cost of eliminating them completely would be higher than the cost of living with them. Your body doesn't try to sterilize your gut. You host trillions of bacteria, some beneficial, some neutral, some potentially harmful. Your immune system maintains a balance, allowing these microbes to coexist with you as long as they don't overstep certain boundaries. It's a negotiated peace, not a total war. And this tolerance might extend to parasites too. We talked about the hygiene hypothesis, the idea that reduced exposure to parasites in modern environments might be contributing to increases in allergies and autoimmune diseases. If this is true, it suggests that humans and many other species evolved in the presence of parasites. Our immune systems expect them. They're calibrated for a world where parasitic infections are common. Remove all parasites and the system misfires, attacking pollen or food proteins or the body's own tissues. In this view, parasites aren't just tolerable, they're necessary. Not because they're beneficial in themselves, but because our biology is built on the assumption that they'll be there. This leads to a provocative idea. Maybe we shouldn't be trying to eliminate all parasites. Maybe the goal should be management, not eradication. Reduce the burden enough to prevent serious disease, but maintain some level of exposure to keep immune systems properly calibrated. It's a controversial stance, especially for people living in regions where parasitic diseases like malaria, schistosomiasis, and river blindness cause immense suffering. But it's worth considering as a philosophical principle. That life evolved in a world full of parasites, and that trying to create a parasite-free world might have unintended consequences. Now let's zoom out even further. Parasites remind us that autonomy is an illusion. We like to think of ourselves as independent agents, making our own choices, following our own goals. But we're embedded in relationships. We depend on plants for oxygen, we depend on bacteria to digest our food, we depend on other humans for language, culture, and meaning. And we're influenced, perhaps subtly, by the parasites and microbes living inside us. The boundary between self and other is porous. You're not a discrete individual. You're a community, an ecosystem, a collaboration between trillions of cells and multiple genomes. And this perspective is humbling. It deflates the ego. It reminds us that we're not separate from nature, standing above it, controlling it. We're part of it, shaped by it, vulnerable to it. A single-celled organism can alter your mood. A virus can end your life. A fungus can take over an ant's body. These aren't signs of weakness, they're signs of interconnection. Everything is touching everything else, influencing, shaping, responding, and we're no exception. But there's also something beautiful in this. The fact that parasites and hosts co-evolve, that they shape each other over millions of years, creating intricate dances of adaptation and counter-adaptation shows how deeply connected life is. Nothing evolves in isolation. Every organism is responding to other organisms, and those responses create feedback loops that generate complexity, diversity, and resilience. Parasites aren't just taking. They're also giving, in the sense that they drive evolutionary change, maintain biodiversity, and create ecological stability. The world would be simpler without them, but it would also be poorer. And this brings us to the question of meaning. What does it mean to be a host? To carry something inside you that isn't entirely you, that has its own agenda, its own evolutionary goals? In a way, we're all hosts. We carry memories, ideas, cultural norms, habits, beliefs, none of which originated with us. We inherited them, absorbed them, and now they influence how we think and act. Are these any different from parasites? They're not biological, but they're still replicators, entities that use us as vehicles for their own propagation. When you share an idea, you're transmitting it. allowing it to reproduce in someone else's mind. Ideas are parasitic in the broadest sense, but we don't usually think of them that way because we've accepted them as part of who we are. Maybe parasites are the same. Maybe the organisms living inside us, altering our chemistry, influencing our behavior, are part of us in a meaningful sense. Not welcomed perhaps, but integrated. Part of the extended phenotype that is you, the full constellation of biological and cultural and microbial influences that create your experience of being alive. This doesn't make parasitism less real or less harmful in individual cases, but it does complicate the narrative. It makes us question where we draw the line between self and other, between cooperation and exploitation, between harm and coexistence. So, as you lie there tonight, drifting towards sleep, feeling your body relax, your mind quieting, consider this. You're not alone. You never have been. You're hosting a multitude, bacteria on your skin, viruses in your cells, maybe a few dormant parasite cysts in your tissues. And all of this is normal. It's how life works. You're a walking ecosystem, and the health of that ecosystem depends on balance, on negotiation, on countless organisms finding ways to coexist even when their interests don't perfectly align. And maybe that's the real lesson from Parasites. That life isn't about purity or independence. It's about relationships, messy and complicated and sometimes harmful, but also creative and generative and essential. Parasites force us to confront the reality that we're always entangled with others, always influencing and being influenced. And once you accept that, once you let go of the illusion of complete autonomy, something shifts. You become more curious, more compassionate, more aware of the invisible threads connecting you to everything around you. So thank you for spending these hours with me, exploring the hidden kingdom of parasites. Thank you for being curious enough to listen, to learn, to sit with ideas that might be uncomfortable or strange. If you've made it this far, if you're still here, still awake, or drifting pleasantly at the edge of sleep, go ahead and leave a like if this helped you. Subscribe if you'd like more journeys like this. And drop a comment telling me what surprised you most, what you'll remember, or what you're thinking about now. I read every comment, and your reflections help me understand what resonates, what works, what to explore next. And now, as we close, take a deep breath. Feel the weight of your body. Notice the softness beneath you. Let your eyes stay closed if they're already closed. And know that the world is far stranger, far more interconnected, and far more beautiful than we usually have time to notice. Parasites are part of that beauty, not in spite of what they do, but because of it. Because they show us that life finds a way, always, even in the most unlikely places, even in the most intimate relationships. The invisible kingdom is vast, and we've only glimpsed a fraction of it tonight. Sleep well, dream of forests full of unseen connections, of crickets walking toward water, of ants climbing grass, of ancient partnerships written in genes. And remember, when you wake, that you're part of this story too, a host, a partner, a participant in the endless dance of life shaping life. Good night. So, here we are, 3 hours later. You've traveled with me through forests and oceans, through microscopic worlds and ancient evolutionary timelines. You've met crickets driven to water, ants locked in death grips, crabs transformed into surrogate parents. You've learned about chemicals that rewrite thoughts, parasites that cross barriers we can't, and relationships so intimate they blur the boundary between self and other. And through it all, I hope you found what you came here for tonight. Not just knowledge, but calm. Not just facts, but perspective. A gentle reminder that the world is vast and strange, and that sometimes the best thing we can do is simply let go and observe. Remember how we started? I asked you if you'd ever felt like something else was making decisions for you, like some invisible force was guiding your impulses, your cravings, your sudden changes in routine. And now, after everything we've explored, that question takes on new dimensions, because the answer is yes. Not in some mystical or supernatural sense, but in a biological one. Right now, your body is making decisions without consulting your conscious mind. Your brain is releasing adenosine, building up pressure towards sleep. Your circadian rhythm is telling you it's time to rest. Hormones are shifting, neurotransmitters are changing concentration, and all of this is happening automatically beneath your awareness. And if you're carrying toxoplasma, if you've got dormant cysts in your neurons, they might be contributing too, subtly altering dopamine levels, nudging your personality by tiny increments. You'll never know for sure unless you get tested, and even then, you won't feel the difference. Because this is how biology works. It's not dramatic, it's not overwhelming, it's just constant, quiet influence, shaping you from the inside out. And honestly, that's okay, because understanding this doesn't make you less you. It makes you more aware of what you actually are. A biological system, exquisitely complex, responding to internal and external signals in ways that mostly serve your survival and well-being. Parasites teach us something profound about control. They show us that consciousness, that feeling of being in charge, is built on a foundation of chemistry. Change the chemistry, and you change the experience. This isn't a depressing thought. It's actually liberating. Because if you understand that your anxiety, your restlessness, your difficulty sleeping, these are chemical states, then you can approach them differently. Not as personal failures, not as character flaws, but as biological conditions that can be managed, adjusted, rebalanced. And sleep, that thing you're hopefully drifting toward right now, is the ultimate act of letting go. Every night, you surrender control. You close your eyes and trust that your body will keep breathing, keep your heart beating, keep regulating temperature and repairing tissues. You enter a state where you're vulnerable, unconscious, disconnected from the world, and yet you do it anyway, because you have to. Because sleep is when your brain consolidates memories, clears metabolic waste, resets neurotransmitter systems. Without sleep, you die. Not slowly, not comfortably. Within days or weeks, cognitive function would collapse, hallucinations would begin, and eventually the body would shut down entirely. So, sleep isn't optional. It's essential. And the fact that you're here, listening to a 3-hour video about parasitic biology, suggests that maybe sleep hasn't been coming easily lately. Maybe your mind races. Maybe you scroll through your phone for hours, stimulating your brain when it needs rest. Maybe anxiety keeps you awake, replaying conversations, worrying about tomorrow. If that's true, I want you to know something. You're not broken. You're just a human living in a world that wasn't designed for human biology. Artificial light disrupts circadian rhythm. Stress hormones flood your system in response to emails and notifications. Your brain is trying to function in an environment radically different from the one it evolved for. But you can work with your biology instead of against it. Dim the lights an hour before bed. Let your melatonin rise naturally. Avoid screens. or at least use filters that block blue light. Create a cool, dark, quiet environment, and give your mind something to focus on that isn't anxious thought loops. That's where videos like this come in. Not to excite you, not to stress you, but to gently occupy your attention while your body does what it needs to do. Relax, release, drift. Over the past 3 hours, we've covered an enormous amount of ground. We started with the puppet masters, parasites that manipulate behavior through neurological hijacking. We explored how they access the brain, how they produce neurotransmitters, how they turn hosts into vehicles for their own reproduction. We met the zombie makers, fungi that guide ants to their deaths with chemical precision. We followed hairworms as they drove crickets into water, completing life cycles that span land and sea. We watched Saculina transform crabs from the inside out, castrating them, feminizing males, turning them into nurseries for parasitic offspring. We examined the chemistry of control, the molecular weapons parasites use to evade immune systems and rewrite neural code. We traced complex life cycles through multiple hosts, marveling at the coordination required for parasites to complete their journeys. We saw how parasites make hosts build structures, galls and webs and shelters designed for someone else's benefit. We confronted the darkest manipulations, parasites that orchestrate suicides, driving hosts to sacrifice themselves at precisely the right moment. We brought it closer to home, exploring parasites in mammals and humans, considering the possibility that toxoplasma or other organisms might be influencing us right now. We looked at the evolutionary arms race, the endless back and forth between parasite and host, each side adapting in response to the other. We appreciated the hidden biodiversity of parasites, the thousands of species living invisibly in every ecosystem, playing essential roles in population control, energy flow, and ecosystem stability. We traveled back through deep time, tracing the origins of parasitism to the earliest days of complex life. We looked forward to the future, exploring how parasites are inspiring new medical treatments, drug delivery systems, and technologies. And finally, we stepped back and asked what it all means, what parasites can teach us about cooperation and conflict, about autonomy and interconnection, about what it means to be alive in a world where everything is entangled with everything else. If you take one thing away from tonight, let it be this: you are not separate, you are not an island, you're part of a web. A biological community that includes your own cells, yes, but also bacteria, viruses, maybe a few parasites, all coexisting in a dynamic balance. And that's not a flaw, it's the fundamental nature of life. We are all ecosystems. We are all collaborations. And understanding that, truly internalizing it, changes how you see yourself and your place in the world. It makes you more humble because you realize how much of what you think and feel is shaped by forces beyond your conscious control. But it also makes you more empowered because once you understand the mechanisms, the chemistry, the biology, you can start working with them. You can create conditions that support healthy neurotransmitter balance. You can manage stress. You can prioritize sleep, knowing that it's not laziness or indulgence, but biological necessity. And it makes you more compassionate toward yourself and toward others. Because if behavior is chemistry, if thoughts are neural firing patterns, if emotions are hormones and neurotransmitters, then struggling isn't a moral failing. It's a biological state. And biological states can be changed, not always easily, not always quickly, but they can be changed. With the right environment, the right support, the right interventions. So, as we close tonight, I want to thank you again for being here, for choosing to spend 3 hours learning about something strange and wonderful, for being curious enough to follow this journey wherever it led. If this video helped you relax, if it gave you something to think about, if it made the night a little less lonely or the silence a little less heavy, then it served its purpose. Go ahead and hit that like button if you enjoyed it. Subscribe if you want more explorations like this. Science, biology, history, all filtered through a lens of calm curiosity designed for late-night listening. And I'd love to hear from you in the comments. Tell me, what surprised you most? Was it the zombie ants? The hairworms? The possibility that toxoplasma might be influencing human personality? Or maybe it was something philosophical, the realization that autonomy is more complicated than we think, that we're all hosting invisible passengers? Let me know, and tell me where you're tuning in from, what time it is, and whether this helped you sleep or kept you awake with curiosity. Your comments help me understand what resonates, what to explore next, and honestly, they're just wonderful to read. Before you go, one last thought. Tomorrow when you wake up, when you go about your day, try this. Notice the invisible. Think about the bacteria in your gut helping you digest food. Think about the mitochondria in your cells generating energy. Think about the trees producing the oxygen you breathe. Think about all the unseen processes, the hidden relationships, the quiet collaborations that make your life possible. And maybe, just maybe, feel a little gratitude for the complexity, for the fact that life is so much more than it appears on the surface. The world is full of parasites, but it's also full of partnerships, of ancient collaborations, of organisms finding ways to coexist despite conflicting interests. And you're part of that story, not separate from it, not above it, right in the middle of it, connected to everything, influenced by everything, contributing to everything. That's what it means to be alive. To be embedded in relationships, messy and complicated and sometimes uncomfortable, but also beautiful and generative and endlessly fascinating. So rest now. Let go. Trust your biology to do what it does best. Your brain will sort through today's experiences, filing away memories, clearing out metabolic debris. Your immune system will patrol, keeping you safe. Your heart will beat steadily, reliably, without you having to think about it. And when you wake, you'll be restored. Ready for another day of being a walking ecosystem, a collaborative organism, a participant in the vast, strange, interconnected dance of life. Good night, sleep well, and thank you for letting me be part of your evening. Until next time, stay curious, stay kind to yourself, and remember, you contain multitudes